Good afternoon, everybody. Madam has joined us, Professor Shagota Dashgupta. So, before Madam presents and uh, gives us her insightful speech, I would just like to introduce her by saying just a few words. Uh, Professor Shagota Dasgupta is a distinguished biophysical chemist and professor of chemistry at IIT Kharagpur. She received her BSc Honours in Chemistry from Presidency College, Kolkata, completed her MSc at the Indian Institute of Technology, Kanpur, and earned her PhD from the Rensselaer Polytechnic Institute, US. Her research in biophysical chemistry spans protein-protein and protein-small molecule interactions, protein aggregation and fibrillation, glycation and oxidative stress in proteins, and nanoparticle protein interfaces, combining fundamental biochemical insights with translational applications such as biomaterials and drug delivery nanostructures. With nearly 170 plus publications, 5,900 plus citations, she is widely recognized for her scientific impact. She is a fellow of the Indian Academy of Sciences and Indian National Science Academy and currently serves as the Vice President of the Indian Academy of Sciences and the Chemical Research Society of India. She also has various patents, awards, and achievements to her credit. She is the recipient of the CRSI Bronze Medal in 2016 and the Darshan Ranganathan Memorial Lecture Award in 2013. An accomplished educator, she has taught a wide range of undergraduate and postgraduate courses, particularly in biochemistry, computational chemistry, bioinformatics, and biophysical chemistry, and has played an active role in designing curricula, introducing new interdisciplinary courses, and strengthening laboratory training modules. She is also known for her commitment to modernizing science education, exemplified by her role as an instructor of the widely attended NPTEL courses on biochemistry, experimental biochemistry, and fundamentals of protein chemistry, which has benefited thousands of students across India and abroad. In addition to classroom teaching, she has supervised numerous MSc, MTech, and PhD students. In sum, she represents a strong amalgam of scientific rigor, interdisciplinary breadth, educational dedication, and translational vision, making her one of the most respected figures in contemporary and Indian chemical sciences. So, thank you, Madam, for joining us and lending your busy time, uh, a part of your busy time to us, so we could be enriched and madam just for the information you will have 80 minutes for your lecture. After that it will be followed by a question answer session or interactive session. Thank you, madam.
Yeah, thank you very much for that very long introduction. And I would probably finish a bit earlier than that so that we can interact a bit more. I found that that is better with the participants and the audience. So maybe a bit more than an hour, and then we can have an interaction session. So, a very good afternoon to all of you, and I hope after your lunch that definitely had a bit of protein in it, you are ready to understand a bit about proteins and their interactions. Now, our interest lies in the proteins, as you know. So, if we look at the, oh, let me just get my pointer up here. So when we talk about the cell, the level 4 of our organization, and then look at how we can break it down, we see that we have from the organelles and the cells the several types of supramolecular complexes. Following this, we have macromolecules such as DNA, protein, and cellulose, and each of them have their own monomeric building blocks. Now, the building blocks are extremely important in getting the specific structure and function, as we will see when we consider our interest, that is with amino acid and proteins. Now, if we consider the peptide bond formation, which I assume all of you are aware of, where we have two amino acids linked together to form this peptide bond, the peptide bond has several characteristics that is extremely important to the overall structure of the protein. So when we link even further amino acids together to form further more and more peptide bonds, we have a structure that is like this, where we have beads connected on a necklace, where we have the beginning of the protein that is known as the N terminus and the end of the protein that is known as the C terminus. Now, as we go along in trying to understand this, we see that this linking of the amino acid together is going to give us a polypeptide chain. Now, when we have this polypeptide chain, a question arises is that do all polypeptide chains form proteins? Now, when we talk about a protein is a polypeptide chain, but it is one that will occur in nature and it will fold into a unique three-dimensional structure. Now, not all polypeptide chains are going to fold into the same structure every time. However, if we have a polypeptide chain that occurs in nature that folds into this well defined three dimensional structure, we will call it a protein. Now, this protein will have several ways of formation or several levels of hierarchy in structure. So, once we form these amino acid linkages here in the primary structure, as it is called, this is the assembly formed by the peptide linkages that link one amino acid to the next. Following this, we have the secondary structure, which is mostly a folding structure given by a specific geometric requirement, and the interactions here are hydrogen bonding. This hydrogen bonding patterns give us either the pleated sheet or the alpha helix, which is a level of folding that we get from the polypeptide chain that forms the primary structure or the sequence of the protein. Following this, we have the tertiary structure, which gives us the packing, the folded, well compact structure. Some proteins also have a quaternary structure, which leads to interactions of these monomeric units to form multimeric proteins. So, given these levels of structure, there are a lot of studies into how we can get from the primary structure to the final tertiary folded structure. And as all of you are aware, the alpha fold that was a predictive mechanism that told us that the primary sequence would form a specific tertiary structure, and this was awarded with the Nobel Prize last year. So, in trying to understand what we know by these structures and how we can look at these structures, we see a folded compact structure that is our protein. Now, in trying to understand what are the interactions and how this protein actually functions depends upon its structure. So, when we're going from the primary to the secondary to the tertiary, and in some cases the quaternary, the whole folded structure is unique in nature. And this unique structure is essential to perform its function. So, this protein folding mechanism that occurs to give the native state of the protein, we understand the importance because if this structure is not achieved, failure to adopt this structure can lead to several diseases. Now, leading to diseases indicates that we have a misfolded protein, and this gives us a series of protein misfolding diseases that are very debilitating and can be fatal as well. So, understanding how the protein folding occurs, understanding how we get the native state of the protein, and the particular interactions that occur is an extremely important factor. So, based on this, there was this beautiful experiment conducted by Anne Finson, where a protein, ribonuclease A, was taken as the model protein. And as you can see, this is the bead of the amino acid sequence. And what we see here is this protein has an additional covalent bond. The peptide bond is a covalent bond that links two amino acids together. Apart from this, the other covalent bond indication of a protein is the formation of this disulfide bond, the cysteine-cysteine to form the cysteine disulfide bond. Now, in ribonuclease A, there are four such disulfide bonds that have been color coded here as pink, yellow, green, and blue. Again, we see the four ones on the left hand side in the active protein, indicating that this is the folded structure. Now, the experiment that was conducted by Anfinsen, he oxidized these. So these were broken up, the disulfide linkages were broken up, and then the protein was allowed to unfold. So now that we have a denatured, unfolded protein, this implied that when this was removed, when the denaturing agents were removed and the protein was allowed to fold, further reduced, then it meant that this particular amino acid residue, the cysteine, could have linked up with any other one similarly for the others. So, but this linking did not occur. What it meant that this pink colored cysteine recognized the other pink colored cysteine, giving us an idea that the linkages that were known by the sequence itself, leading to the famous statement of Anfinsen that the essential structural information is stored in the primary sequence of the amino acid. So this is the primary sequence, and when it is allowed to fold, it will fold into this unique structure only. Now, this was a very profound statement which also won him the Nobel Prize. So, if we look at the general factors of protein folding, we have the amino acid sequence, and now we understand that this amino acid sequence determines how the polypeptide chain is going to fold into its three-dimensional structure. So what we have is we have a large number of conformations possible, indicating a large entropy. And as we, this is called the folding funnel. So as we sample different type of structures, we get to a partially folded structure, usually referred to as the molten globule. And then we have the native state of the protein, that is the unique three-dimensional structure. The important elements for protein folding, therefore, are the sequence of the protein, which indicates the specific peptide linkages and the cysteine-cysteine disulfide bonds. The specific cellular environment, the temperature, the pressure, the pH, then a balance of the various folding states that is going to get us from the unfolded state to the folded state. Following this, a fully functional protein quality control system. This is a beautiful way in which the body works. A quality control system that uses chaperones, that are other proteins that assist in the folding of a protein if a protein happens to misfold. Because we know that a misfolded protein is going to lead to disease. So there are systems in our body, the proteasome unit or the protein quality control system, that looks for this misfolding, looks for these misfolded proteins, allows them or gives them an opportunity to fold correctly, following or failing which they are marked and discarded in the system. So if we look at all these systems put together in the protein chemistry laboratory at IIT Kharagpur, as was mentioned in the introduction, we work on several methods related to protein chemistry, whether it is protein inhibitor design and development, protein fibrillation or aggregation studies, oxidative stress and glycation, or now recently we've been working with waste proteins for several diverse applications. So the questions that we try to answer is, if I have a knowledge of the structure of the protein, do I know its function? Can I list the interactions? Can I understand the binding of the protein? So these are all the questions related to a protein structure and how we can go about doing it or understanding it. The first thing that we look at is protein ligand binding. Now when I look at, because any interaction involves a binding. Now, when I have a ligand, this is typically a small molecule, but obviously we can have protein-protein interactions, protein-DNA interactions, where we will have two macromolecules actually interact with each other. It is important to know or have a knowledge, therefore, of where the ligand is going to bind. Is there an active site? Is there a specific region that the protein or the ligand is going to bind to? This means that when I look at specific binding, for example, the most common example will be an enzyme substrate complex. There, I would be looking for steric complementarity, geometric complementarity, and chemical complementarity. In this fashion, I will be able to know whether my ligand is going to fit into my protein molecule. And based on that, I will have different aspects of research that I can get to. One is basic research, which is done to increase the knowledge and understanding from an academic standpoint, whether or not the specific ligand that I'm interested in is going to be bound to the specific protein of interest. So that would form basic research, extremely essential to get on to applied research, where this is done to solve a problem related to the system of interest. Following this, you have now technological development that takes a consideration to improve the quality of life. But without basic research, you cannot get to applied research, and without either of these, you can get to technology development. So people look at technology readiness levels now in research activities, but without the basic knowledge, without the basic concepts, it is difficult to consider this. So in the problem definition, there are two systems that we consider here. One, of course, is the protein of interest, and the other is the specific ligand of interest as well. So a series of ligands and a specific protein, or a series of proteins also, that give us information, known as host-guest interactions. Now, because this was a statement by Linus Pauling that tells us that the secret of life is based on molecular recognition, where it is the ability of one molecule to recognize another, and these molecular interactions are of extreme importance to consider the build-up or the understanding of how proteins interact with ligands, how proteins interact with other proteins, and how proteins interact with nucleic acids or even amongst themselves. So this understanding is very important, and the example that I'm going to give you now is going back to enzyme action and inhibition. Once I give this brief introduction, I will be going to some research activities that we have been conducting over the years to show you how we have implemented these basic ideas in our research. Now, when we talk about enzyme action and inhibition, all of us know that enzymes are biological catalysts. What does this mean? It means that it lowers the activation energy barrier, in the sense that it is going to speed up a specific biochemical reaction. They do not change themselves, and they reduce the activation energy. So any of the reactants going to the products following this high energy activation barrier without the enzyme is going to have a reduced activation energy with the enzyme. And it is this experience or this idea that we exploit to understand how the enzyme works. So when we talk about the enzymes, they are extremely specific in nature. So if we consider an example such as sucrose that binds to this specific enzyme, we see that the orientation of the molecule is extremely important, because we understand that the 6-membered ring will fit in this pocket and the 5-membered ring is going to fit in this pocket. So the specific active site and the specific formation of the enzyme substrate complex indicates that there is a specific orientation, a specific recognition. So this substrate fits perfectly with the enzyme. Now, another thing very important in enzyme substrate interactions is that the enzyme substrate binding cannot be too strong, because if it is too tight, then the enzyme is not going to allow for any reaction to occur, and it is not going to allow for the release of the products. So it has to be optimally energy efficient so that it will bind the substrate, perform the specific reaction, in this case the hydrolysis of this bond, creating the glucose and the fructose, where the products will be released because this is not so tightly bound, and you would get the enzyme back in the system, which can then again be used for the binding of another substrate molecule. So, this beautiful way in which enzymes work is what is extremely important in trying to understand how you design inhibitors. Now, when you design an inhibitor for an enzyme, unlike the substrate binding being relatively weaker, you want the inhibitor to have an extremely strong binding with the enzyme so that it cannot react or it cannot perform the function. So the enzyme is rendered inactive. Now, this idea of how you would design a molecule to sit at the active site of the protein indicates that you need some more knowledge about the substrate binding specificity. You need a bit more knowledge about the enzyme and more knowledge about the substrate. So once the substrate sits in this specific active site, there are certain interactions that occur. Now, when we look at these specific interactions, there are specific modulations that occur in what is called the lock and key model or the induced fit model in allowing the substrate to bind to the enzyme. But in each case, you will observe that the active site of the enzyme is made available exactly the same way after the product is released so that it is ready to bind another substrate molecule. So this factor is extremely important. And when we talk about enzyme inhibition, they are compounds that are going to decrease the activity of the enzyme, and most drugs act as enzyme inhibitors, indicating that we need to know the structure of the enzyme, we need to have an idea of the enzyme, and then decide upon how important these inhibitors are in determining the enzyme mechanisms and how we can look at the nature of the active site to know how the inhibitor works in what is called drug design, which I will give you an example of. So, when we consider the topographic surface or the geometry, the binding pocket, we look at our specific enzyme binding site. So, if this is the binding site and we have a specific molecule come and sit in the binding site, what we need to know is we need to have more information about the binding site if I have to design a molecule that is going to be interacting with this setup, at the same time forming interactions that are more difficult. So I look at it in a bit more detail and I find out that there are specific amino acid residues here where I have certain interactions possible, whether they are hydrogen bonding interactions, just hydrophobic type, ionic interaction type. I will have therefore my substrate molecule come and sit in the binding site. Now, we can have designs of molecules that would increase these interactions, increase the capability of this particular substrate analog to come and sit at the active site and have no reaction occur if I were to create an inhibition for the specific enzyme. So in these ideas, if we take these ideas ahead, I will tell you the proteins of interest that we have been working on for the past 3 decades, where we have looked at human serum albumin, ribonuclease A, angiogenin, lysozyme, gamma crystalline. So these are the proteins that we have been actively working on and looking at their structural aspects, looking at their interactions, and so on and so forth. In protein inhibitor design and development, we look at a process called angiogenesis. Angiogenesis is a process whereby that there is a growth of blood vessels from an existing vasculature. What does this mean? This means that there is a blood vessel and there are certain cells, for example, in this case, tumor cells. And if there is a release of angiogenic factors, these are called growth factors. Now, what happens, for example, if we do not go into tumor cells or angiogenic factors right now, and we consider that we have a cut on our arm or hand. Now, what happens is this cut, this wound heals. That means there are some growth factors that are involved in healing the wound. These are called angiogenic factors because you need the growth of blood vessels to heal the wound. However, interestingly, it is the same growth factors that are also involved in tumor growth. But every time we get a wound, we do not get a tumor. So that means there is an extremely delicate balance between the anti-angiogenic factors and the angiogenic factors. So the tumor growth occurs with this blood vessel and with the process of angiogenesis. Now, this means that if we can recognize or control angiogenesis, this could lead to cancer therapies, which has resulted in extensive research in this particular field. So, what happens is when we have these angiogenic factors, they stimulate the formation of new blood vessels from pre existing ones. And as we see, they play a very important role in physiological and pathological processes. So, we can have angiogenesis or anti angiogenesis depending upon whether we have a greater amount of angiogenic activators or angiogenic inhibitors. Now, this is important because the several growth factors that are known, there is the acidic and basic fibroblast growth factor, there is a transforming growth factor, TGF alpha and beta, and angiogenin. Now, angiogenin is actually a small protein. Now, when we look at angiogenesis, the excessive angiogenesis causes disease. Again, insufficient angiogenesis will also cause disease. So, ideally, this protein can be a molecular target where we can look at the activity of the protein and see how we might inhibit the activity of the protein. And the good thing, or the rather advantage of this protein, is that it is a member of the ribonuclease superfamily. What does that mean? It means that the ribonuclease A, which is a very big ribonuclease family, has a series of ribonucleases. And what we mean by a ribonuclease, for example, ribonuclease A, it is a protein that is going to degrade RNA. Now, angiogenin, being a member of the ribonuclease superfamily, also has the ability to degrade RNA. And this is the enzymatic activity of angiogenin. In addition to the enzymatic activity, it also has a biological activity, that is the buildup of the blood vessels. So, ideally, what would happen is if there were an in situ tumor and there were specific angiogenic factors, the capillaries would grow with the angiogenesis factors. So, we have angiogenic proteins that are going to be released, forming this big tumor. Ideally, if you had anti-angiogenic drugs, then the protein would be able to diminish the tumor. So that is where the question lies. So we have the protein angiogenin. It is a small protein with 123 amino acid residues. And as I said, in addition to its ribonucleolytic, that is enzymatic activity, it has the biological activity of creating blood vessels. So we used, or we isolated angiogenin from blood as well as express the protein. So we have two sources of angiogenin which we have used for our experiments. Now, when we isolate, I will give you a brief background of the isolation of proteins. So, when we isolate proteins, we have them, since blood is a tissue, there are several ways in which we have to isolate them. So, what we have to consider is if we take the cell homogenate, we have to resort to a low speed centrifugation, where what happens is the pellet here will take in the larger particulate matter here that forms the whole cells, the nuclei, the cytoskeletons. So you want to go gradually as you go along, because if you go for high speed centrifugation right away, everything is going to precipitate. But you are interested in the macromolecules or the proteins which you will finally isolate and get the purity of and characterize. So here we have now the so we take now the supernatant, and subject this to medium speed centrifugation. And as you can see, we have again the larger particulate matter come to the bottom of the test tube, which primarily consists of mitochondria, lysosomes, and peroxisomes. Following this, we go to slightly higher speeds and then higher and higher speeds till we get our level of interest. Then, following this, we have to take the specific supernatant and separate it. Now, in the chromatographic methods that I use, we have gel filtration chromatography that acts as a molecular sieve where the proteins pass over a column that is filled with porous beads. We will look at a bit of it. Then we have ion exchange chromatography where you exploit the charge on the protein, and you have affinity chromatography that exploits the affinity to a specific type of ligand, be it glucose or DNA or some such ligand. So we have these different methods in gel filtration chromatography, as you can see. So we have our column, and this you can see is the protein sample. Now the protein sample is actually a mixture of three different proteins. Now what is present in the column is something like this, that is zoomed in on the right hand side. We have these carbohydrate polymer beads. Now these polymer beads are a network of polymers, and what happens is there is some aqueous space within the beads. Now once the three proteins, in this case we have three proteins of different sizes, are put into the column, and we have what is called an elution of the column. That means we are bringing down the protein, whether due to gravity or due to some pump action. We are considering the variation in these beads. What happens is the smaller beads get trapped in the polymer network of the bead, in the carbohydrate polymer network. The medium size are sometimes partially trapped, but the large ones can evade the beads and they do not get trapped and they come on the outside. So what happens if this is the specific flow direction? We will have the larger beads elute first, followed by the medium size, and then the smaller size ones. So as we separate the, or we elute the column, we will see there is a gradual separation. And what will happen when we check the UV absorbance of each of these test tubes? The one that is going to have an absorbance at around 280 nanometer indicates the presence of the protein. So a series of these test tubes will be collected, and knowing that the larger protein is going to come out first, we will be able to find out what the molecular weight is. But before that, what we need to do is we need to also understand about ion exchange chromatography. In this case, what we do is we exploit the charge on the protein. For example, in this case, you also have beads, but the beads themselves now have a charge to it. So if we have a negatively charged bead, what is going to happen? The protein mixture, in this case, the blue and the pink one, you can see that the blue has a greater positive charge than the pink protein, which indicates that the negative bead is going to attract the positively charged protein bead to a greater extent than the other one. So now, based on the charge of the protein, we have a separation. So each of these methodologies in chromatography exploits some property that is going to allow for the separation. Then we look at affinity chromatography. In affinity chromatography, for example, in this particular case, if we have a protein that is attracted to glucose, or glucose binding occurs, so we have a specific bead that has tagged to it glucose. Now what is going to happen if we have a mixture of proteins? The protein that has an affinity for glucose is going to get attracted to the glucose and it is going to bind to the glucose, whereas the other protein is not going to bind. So now what is going to happen? If I add extra glucose to the column now, it will release themselves from the bead and attach themselves to the other glucose molecules and then be eluted. So these are three different ways in which we can get our protein of interest. The chromophores are the specific chemical group which you are aware of that absorbs light of a specific frequency. And in this case, if we look at a way, if we want to find out whether we have a protein, we can have a specific assay called the Bradford assay that will tell us whether our solution, because if we have a solution of the protein that has been so-called eluted from the column, if it is not a colored protein, we will not know which test tube has the protein. So we exploit the chromophoric moieties of the specific electronic transitions and look for the UV visible region, which is for aromatic residues that are present in the protein, gives us a signal. So if we have, most proteins have any of these aromatic amino acid residues. So they are going to give a signal if they are present in the protein. So what we will have is we will have tryptophan, tyrosine and phenylalanine. And if we have tryptophan, the tryptophan extinction being higher, it will give us an indication that we have the presence of a protein. So all of the test tubes have to be subjected to a UV test to figure out whether we do have the protein present or not. So now we have separated our protein based on the size, and we are going to look at size exclusion chromatography, and we have a standard. Because we need to know the molecular weight, we use a standard. Now, as you can see, there are 5 different proteins here and 1 vitamin, with 4 proteins and 1 vitamin that have been eluted according to size. What does this mean? It means that the largest protein, that is thyroglobulin, is coming out first. So finally, we can find out a specific KAV value that is given by the specific formula, which I will not go into the details of. This is a typical biophysical problem that we can work out, where we are looking at what is called the void volume, the total volume. But what we are interested in is we are interested to find out the molecular weight. So given the known molecular weight and the KAV values, I can find, or I can get a calibration plot. Then from the unknown protein on the same column and eluted under the same conditions, what I can do is I can find out the KAV value, and from that I can find out the molecular weight of my unknown protein from the standard calibration curve. So this is the way the molecular weights of proteins can be determined. Another way is with sodium dodecyl sulfate polyacrylamide gel electrophoresis, extremely common method used in all protein chemistry laboratories to find out the molecular weight of a protein. So in this case, what we have is we have specific lanes in which we have, so this gel again is a network where we have the protein distribution occur because of molecular weight. Now, what happens is we have the lighter proteins being able to move through the gel faster, which indicates that the decreasing molecular weight or the smaller molecular weight protein will be at the bottom of the gel. So based on this, we also get a calibration curve where we use what is known as a molecular weight marker that gives us an indication saying that here is the lightest protein and we have the others known molecular weights from which we can find out a calibration curve, and from the relative migration of the unknown protein we can decipher what the exact molecular weight of the protein is and see if it is our protein of interest. So following this, we look at the purification of human angiogenin, the protein I was talking about. So we used a size exclusion chromatographic profile for human angiogenin. We had the specific standard, and from the specific standard and from the gel electrophoresis, we determined that we have a pure monomeric protein, indicated by the single band that we see marked with the red arrow here. So this methodology told us that we have the human angiogenin isolated from our setup. Now, I have to find out, or we have to determine whether the human angiogenin that we have is indeed angiogenin. This means I have to do what is called a specific enzymatic assay. What is this assay? This assay tells me whether I have my enzyme or not. Now, knowing that angiogenin is a ribonucleolytic protein, it has the capability to chop up RNA. So, what we do is we take human angiogenin and then take RNA, and this chops up RNA into the RNA fragments. And then, how do I look at these fragments? I look at these using what is called an agarose gel. Now, what you see here is you can just look at the relative intensity. This is RNA. So, since this is not fragmented, I have a high intense band here. Now, when I use human angiogenin, what it has done, it has chopped this up into pieces. So, the intensity has decreased dramatically. So, based on this, I can say that yes, I do have a protein that has the ability to chop up RNA. I won't go into the details of the other ones. Another example or another specific test is whether angiogen has the capability of building up the blood vessels. For this, we have to do or we have to conduct what is known as the CAM assay, because I need a growing cell. This growing cell is given by this chicken egg. So we get eggs from the poultry, and because from the hatchery, not the poultry. The hatcheries provide the eggs because they have to be growing cells. Poultry eggs are treated so that they do not fertilize or they do not hatch rather. So what we have here is, if you can look carefully, you will see a plastic disc here. Now what we do is we take this plastic disc, and on the disc we put the protein, or we put any other compounds that we want to test that can actually be involved in angiogenesis or not. So based on this, we have a control, and since human angiogenin is capable of building up new blood vessels, we see more blood vessel formation, more they're called branching and so on and so forth, more number of junctions. But there is a level that tells us, because the cell itself is also growing, we have to check it with the control. So there is the control growth and there is the human angiogenin growth. But this tells us, since we have a higher number of junctions, higher number of blood vessel formation, it tells us yes, we do have human angiogenin isolated. So based on this, we looked at this specific question, and our idea is, now can I inhibit this? So right about this period, there was this paper that came out in Nature that said that angiogenesis was inhibited by drinking tea. So what we did is we looked at green tea polyphenols and saw whether they were capable of inhibiting the angiogenin induced angiogenesis. These are the major green tea polyphenols known as ECG and EGCG, and they have limited bioavailability and lower aqueous short life, but I will not go into the details here in how to look at this particular methodology. But what I will tell you is the experiment that we conducted with the CAM assay, that is the egg assay. So here you see the control blood vessels. Now with angiogenin, as expected, there is much larger blood vessel formation. However, with the green tea components, you see there is narrowing of the blood vessels compared to the control. And then when we take the angiogenin and the green tea polyphenols, together what happens is they form the thickness according to the control. So this gives us an indication that this green tea components can actually inhibit angiogenesis. So we look at different ways, and as we looked at the intensity of the bands, you can see, since this is only RNA, we will find this most intense. This is the protein, indicating that it has chopped up all the RNA. Now if any intensity shows up, it means that some RNA is still left. So it means that whatever has been added here is able to inhibit the protein so that it cannot chop the RNA. So I have a higher intensity. And as we can see from this, that EGCG has a better inhibitory property than ECG. We did several studies related to this in terms of forming it with nanoparticles and so on and so forth, which I will not get into the details of. but we did see that we do have an anti-angiogenic effect. So this idea said that, okay, here is my protein. I have RNase A and I have angiogenin. You can see they both look very similar, and the parts that are marked in blue are the active sites of the protein. So this is a ribonucleolytic protein. This is also a ribonucleolytic protein. Then what we have here is we have an additional nuclear translocation site and a cell binding site which is absent in ribonuclease A, because this has additional biological property. So this is why we use this protein as a model protein. And based on this, in collaboration with Professor Pathak of the Department of Chemistry, we designed inhibitors, because now we know what the pocket looks like. So we designed specific inhibitors to look at their ribonuclease A inhibition. And since both of them have this activity, we get to the design and development of synthetic inhibitors of ribonuclease A and angiogenin. So these were synthesized compounds. But if you want to go further, there are ways in which you can look at it even computationally, where you have a specific binding site and you look at the ligand molecule sitting in the binding site. And based on this, what we have is we have the structure of the protein. Now, in this case, what you have in the protein data bank, there are structures of proteins, and you can get small molecule structures that will tell you how you can fit a specific molecule in the active site. So if we look at how this protein is rotated, we have the protein structure. So we have an idea of how we can look at the active site of the protein and how the molecule is interacting. So what we can do is we can build up other specific molecules that will tell us how to design a molecule. So we look at the details of the amino acids, as we can see here, the details of the amino acid that line this pocket, and then try and find out how we can work on this molecule or any similar molecule to find better and better inhibitors of the enzyme. So this is the whole idea of designing an inhibitor for a protein that takes it further into trying to understand the disease conditions, in this case with angiogenin. So this idea tells us that if we have a whole ligand database built on the computer and we have a target protein, we can do these docking methods, as they are called, into the active site of the protein and then get the information about how the rational design can be taken further through computer modeling. And once we even all of these specific software give you a score. So once we have this specific score, we can then decide upon whether this molecule should be synthesized. And now you hear about AI in drug design. This is exactly what AI does. But of course, you would need somebody to make the molecule for you. So the chemist is definitely required for the synthesis procedure, the testing procedure, to find out whether the ligand or the specific small molecule actually works on the target protein. And so when we look at drug discovery, design and development, there are many, many compounds that are started off with preclinical development, clinical development, that finally gets to that single compound that we will call a drug. So this is where artificial intelligence in drug design comes up in a big way. Now, the second story I'm going to tell you about is about protein modifications in oxidative stress. I may not talk about glycation, but tell you about oxidative stress. Now, when we talk about oxidative stress and biomolecules, we know that we have proteins, we have membranes, we have DNA, we have ligands. Now, all of these oxidative stress mechanisms work, say, through UV light, ionizing radiation, and different ways in which we can have these free radical formation. Now, free radical formation can result in a lot of protein damage or DNA damage or membrane damage. Now, all of these are going to create problems. And what kind of problems can they actually create? There are lots of diseases that are caused due to oxidative stress. And there are several ways in which they can also be detected. For example, if we have cataract formation, which I will tell you a bit about in a moment, there is one amino acid we all know about, tryptophan. Tryptophan oxidation occurs when cataract formation is occurred. Then we can have histidine, tyrosine oxidation. So these methodologies tell us that something is wrong in the disease conditions. And the oxidative species, that there are specific reactions in our body that require the free radicals. But you also have to have an anti-radical free radical mechanism that is going to take care of these. In addition, there are external sources that are causes for oxidative stress. And as I mentioned, there are cataract conditions, cardiovascular, Alzheimer's disease, rheumatoid arthritis, all caused due to some radical formation and the fact that the radical destruction does not take place in the normal way. Now, what happens when there is oxidative stress in proteins? We have a protein. What can we have? We can have backbone oxidation. We can have the breakdown of the protein. We can have side chain modification. We can also have protein cross linking, where we will have proteins come together in forming an aggregate. So, if we look at backbone oxidation, there are specific ways in which they interact, specific ways in which they act. We can have protein fragmentation, side chain modification, and cross-linking in what is called dityrosine formation, where we have two monomeric proteins come together to form a dimer or even higher oligomers. So, when we look at these specific dityrosine linkages, there must be a way by which we can measure their formation. Fortunately, the dityrosine acts as a fluorescence probe. So what happens, I can have this fluorescent probe that will tell me whether dityrosine is actually formed or not. So this gives an indication of the extent of the disease, and it is an extent, or rather used as a biomarker, as these are called, of oxidative stress, aging, as well as disease. So these are different ways in which we can study the dityrosine bond formation in the different protein in ribonuclease A that we have looked at, which I will not go into the detail of for now. But the question is, can we prevent this oxidative stress? So we go back to our antioxidative methods, and the common, we are always interested in dietary polyphenols. So the common source of antioxidants are fruits, vegetables, green tea, and so on and so forth. So we use these specific methods. So we have the oxidation, the structural modification of the protein due to oxidative stress. We have an aggregated protein because these are involved in several diseases. And in the protein aggregation study, the disease that we are interested in is cataract formation. So in the last line here, on the last row here, you see cataract formation that is due to the proteins called crystallins. Now there are three major types of crystallins, alpha, beta, and gamma. And the gamma crystallin is the one that is involved in cataract formation. Now this is the inside of an eye lens. What you will see is the beautiful way in which there is an arrangement of the layers and layers of protein. So, if we look at the anatomy of the eye, this is the eye lens. Now, the eye lens has to maintain its transparency throughout the lifetime of the individual. If there is any disruption in this transparency, what is going to happen is there is going to be some aggregation of the protein. For example, this is how the layering occurs. This is the inside of a human eyelids. You can see how beautifully layered the proteins are, and this protein concentration is the highest anywhere in the body. There are layers and layers, as you can see, of the cells that you see here, the layers and layers, and there are around 20 to 25,000 such layers. And any disruption in this ordering is going to result in formation of cataract. So what do we have in cataract? You have the normal lens that is transparent, and cataract that is, you can see it is whitish in color because of the aggregation of the protein. So what happens due to this aggregation of the protein indicates that when you have the light fall on a normal lens, you will have the image formed here on the retina. They converge at the retina. But what happens when you have a cataract formation, there is going to be complete scattering, indicating blurry vision. And what we have seen is how UV radiation affects the proteins that are present in the lens. So if you look at visible radiation, this is expected to go to the retina. But what happens with UVA radiation, it stops at the lens. That means this UV radiation is extremely damaging through the proteins that are present in the eyelids. Based on this, we look at oxidative stress to see what variations have occurred in the protein. Now, if we look at the specific cataract formation, there is cataract surgery that occurs, and in cataract surgery, there is a small incision formed in the eye. The lens capsule is kept as it is, and this protein mass is emulsified and aspirated, and it is thrown away. So what we do is we bring back this protein from the ophthalmologist and see what variations have occurred. And what we see is the variation that has occurred is that a tryptophan amino acid residue has been oxidized to what is called NFK. That means that if I have ordinary protein, then the tryptophan is present. But in a cataractus or damaged protein, I will see the formation of the tryptophan oxidized product. So that means it gives me an idea as to how I can look at it. So here we have the cataractus eye lens. So we looked at the specific spectra, and what I see is I see that I have tryptophan and also I have NFK, that means oxidized tryptophan due to UV radiation. So what we did was we did an experiment where we looked again at EGCG, that is the green tea polyphenols, to see whether we can have the inhibition of oxidation. And indeed, yes, EGCG was able to inhibit the. So here what we did is we took H2O2 and forcefully oxidized the tryptophan and found out whether EGCG could inhibit this oxidation. But that actually did not give us a right idea about the normal protein, because this protein was already a damaged protein because it was cataractus protein. So we have to prepare the protein. So one of my students went for UV photodimage of human gamma B crystalline. So we expressed the protein, prepared the protein, and looked at the effect of the EGCG on the UV damaged protein. So what I just want to walk you through this is we have the native protein. Now when this is irradiated, there is a signal that occurs red shifted. That means you see a damage. So this is at 328 nanometer, that moves to 343 nanometer when I just shine UV light on the protein. Now, if I mix this with ECG, EGCG, or these are the green tea polyphenols, what do I see? I see that the extent of this that was 50 nanometers is now reduced to 3, 8, or 12. So that means that this is able to inhibit the UV radiation damage to the tryptophan. So these are different measures that can be used to understand the proteins, understand ways in which we can look at proteins. So what we have looked at is we have taken the crystalline protein, we have shone it with UV radiation, we have looked at it at low pH, we had looked at it at elevated glucose and seen protein damage. So these are the different ways in which protein damage occurs for crystalline. And in each of these cases, what we have been able to do is we have been able to show that with green tea polyphenols, we can actually have a measure to diminish the environmental stress caused by the damaged protein. So this is just a summary of, so this was a thing that was highlighted in Nature India. We were looking at the RNA complexes. As I told you, the RNA is a type of protein, angiogenin, to see how the interactions occur, and the design of a small molecule in this case said that we have promising drug candidates for a wide range of cancers. This was a bit of a looking at the aids in the proliferation of the cells, as I showed you in angiogenin. In case of the green tea polyphenols, And in terms of cataract formation, our work was highlighted in the Hindustan Times, where it said that green tea can control cataract. That is a huge statement. That is not the case. But we know that what happens is that with the green tea, because of its antioxidant compound presence, what can happen is the harmful effects of the UV radiation can actually be combated, because then what will happen is, we know that UV radiation is a cause of cataract formation, and we can have the scavenging of the free radicals so that we have a better way to look at this. Whatever I presented here would have not been possible without the help of my wonderful students that I have had over the years, my collaborators, and of course, generous funding from a variety of funding agencies. Finally, this is mostly for what all of us need to do, that understanding protein ligand binding is essential for the design and development of compounds and drugs. So this is the take home message. And skill upgradation is looked at in a big way now. So in upgrading your skills, we have to not work harder all the time, but probably work smarter. Thank you for your attention.
Thank you, madam, uh, for your insightful speech, and you have explained it so well.
Thank you so much.
So now the session is open for the questions, and there are quite a lot of, I can see very many thank yous from the participants.
Thank you so much.
Well, I have a question here.
Sure.
Ajoy Kumar Das, he has written, Madam, I have extracted certain peptides. Can you provide some information for its characterization and identification?
Okay, with peptides, basically the size of the peptides is also going to be an important feature,
Yes,
because
madam.
in this, let me just stop this. What's this? Stop projecting. Okay, so what you have is, when you have a peptide, there you have different sizes of peptides. So the first thing that you would need to do is probably do a molecular weight determination to find out maybe a MALDI or one such methodology that will tell you what is the molecular weight of your peptide. So that characterization is extremely necessary. Or what you would have to do is, you may have to sort of do a, like, about an Edmond degradation, where you would, if you want to find the sequence of the protein, then
Excuse me, madam.
yes.
Madam, actually, we have extracted that peptide by using 3 kilodalton cutoff.
Okay.
So, yes,
So
means
it is quite small actually.
it's quite small. And
Right.
just
Do you
we
do
are,
you know do you know do you know what the sequence? You do not know the sequence of
I
the.
don't know, we don't know, madam. Just we have extracted by using that 3 kilodalton cutoff. Now
Mhm.
I am searching for that identification, characterization and
But
identification.
the thing is
Identification.
that if it so happens that your particular peptide does not have an aromatic residue, then
Yes, ma'am.
even with the normal UV methodology, you are not going to get a signal,
No,
right?
we are searching for that LSMSMS, but
Yes. So that was some mass spectrometry you would have to do, like if you do MALDI or LCMS or what, and then
for protein it is fine, but for peptide means nobody is ready to means do that work in our laboratory. It is no, there is no LCMSS. I am from Guwahati, Assam,
mhm.
but in IIST and in NIPAR it is there, but they are not doing that type of work, peptide work. They are
Okay.
doing
There
for
are
protein.
there are some companies who will do peptide sequencing for you. There are
Yes,
some companies in Hyderabad and Bangalore. They do
can you
peptide.
suggest, ma'am? Can
I
you?
won't know offhand, but if you do a search, they will do. There are some that do peptide sequencing for you. So if you send them the peptide, for
Yes,
obviously
ma'am.
payment basis,
Yeah.
there are some companies that do this. So they will give you, or they will tell you what the sequence of your peptide is. But you have to send them, like, you'll have to show an HPLC profile where it is pure. and
Yes,
not a
yes, yes.
mixture of peptides. So if you give them that, there are some companies that do it. Because I remember I don't we because we bought a peptide from a company, but I
Yes,
don't
ma'am.
remember the name right now. So if you send me a mail, I will forward it to my student who wrote to the company. So
Yes,
maybe
ma'am.
okay, you can send me an
That will
email.
be that will be helpful
Sure,
for me
sure.
because searching for a couple of months, but we are not getting.
No, no, sure.
I
You send it to me because I know this company, they make peptides, but I'm
yes,
sure
ma'am.
they will also do some characterization of peptides also. So they will tell you because we bought a pentapeptide from them. So
Yes,
your
ma'am.
molecular weight is probably close to that or even. Okay.
Yes.
So
Okay, ma'am. So can I have your mail ID, ma'am?
it was at the bottom of my lecture. It is SWAGATA
SWA. So
Swagata at the rate of CHEM.
SW small, no?
Yes, small. SWAGATA
ZATA
at the rate of CHEM
at the rate of CHCM.
dot IITKGP dot AC dot in.
Dot
Sir, if I may say, you will
IIT.
also find it in the abstract book. You will find her,
Yeah.
madam's email address in the abstract book as well.
Okay.
Okay, you can send me. Please introduce yourself saying that it's from this lecture. Otherwise, I get so many emails. And I
Yes,
don't.
yes, yes. Okay, ma'am. Sagota at the rate cam.iit.ac.in. IITK.
KGP, KGP,
Yes,
IIT
Kharagpur. Thank you, ma'am.
Yes, sure.
Thank you. It
Since
was a nice lecture, ma'am. Anyway, very good lecture.
Thank you.
next question is from Namrata Ray. Since Namrata Ray is here, you can directly ask your question.
Sure, sure.
Okay, sure. Ma'am, thank you for a wonderful lecture. And I was just writing in the chat box that all great researchers are not great teachers, but you are someone who is both. Thanks a
Thank
lot for your lecture.
thank
And
you.
what I wanted to ask is, ma'am, that you mentioned about dityrosine formation, which is an indicator for oxidative stress. So, does it happen in all proteins or depends upon the side chain? I mean, is it a standard marker for oxidative stress for any protein or?
Currently people are using it as a biomarker, but the way we do it is, since we are looking at induced oxidative stress, because our idea is to see how we can combat oxidative stress. So what we do is we induce oxidative stress in the proteins that we have done so far. We have got dityrosine, but what we need to know is, say there is a single tyrosine and it is buried in the protein. Then the ones that we have, there are six tyrosines and there are three of them on the surface. So what happens is we find that dimer formation occurs when this because it is a CC covalent linkage that occurs. It is a radical reaction, and then we have a CC covalent linkage. So we use that as a marker to see whether the dityrosine has formed. And this is and but nowadays people are using dityrosine as a biomarker for oxidative stress.
Okay, thank you, ma'am.
Next, we have Nobonita Ghosh. Nobonita, are you there or will I ask your question? Okay, Nobonita has asked a question. Madam, can you please elaborate the CAM assay?
Okay. In the CAM assay, first of all, as I said, you have to get the eggs from hatcheries. Okay. So once you get the eggs from hatcheries, then what you have to do is, this is a standard protocol where you have to actually make a hole in the egg and then aspirate a bit of it. So this membrane falls off from the shell, and then you have to actually make a hole in the shell and then tape it first and incubate it for, say, a day or so. And then once you open this sellotape that we put on the egg, we place our plastic disc. So this plastic disc, we create a series of plastic discs, and on the plastic disc we put either the protein or the compound or a mixture of both, or in different concentrations. And then what you do is you invert the plastic disc on this membrane, and then you have to seal the egg and put it in the incubator for 3-4 days, because you need the blood vessels to form. So now after these blood vessels form, then after, it depends upon, so you kind of check it. So after 5 to 7 days, it is enough to take, then we take the picture, but just by that time, when we used to do it with digital camera. Now with mobile cameras that are very good, you can also take it there. So this is a typical standard procedure. The CAM assay is a standard procedure which you have to do to check angiogenesis, because you need a growing cell. So that is the control.
Next, we have a question from Orijit Das. Uh, Orijit, are you there? Okay, let me ask the question. Can we modify a particular active site of an enzyme to inhibit its action or make it bind to a different substrate?
Yes, sure. So that's the whole basis of enzyme inhibition. What The way we look at enzyme inhibition is we make it, we wouldn't call it a different substrate, but we will call it a different compound, where have the idea that you want this compound to go and sit in the active site, but not perform any reaction, so that the enzyme active site is inhibited, and it will not be able to bind any other substrate. And changing an amino acid at the active site would depend upon the amino acids present in the active site, which can be done with site directed mutagenesis. But then again, you have to be careful that any amino acid say change that you do may have also effect on the structure. So you have to be very careful that when you are making this particular change, it does not affect the overall folding of the active site. So that has to be taken care of.
Thank you, ma'am. Rakhi Chakraborty, some of the microphones are not working, so I'm not. Directly asking the questions.
Sure.
Uh, is there any specific dosage of green tea intake that can help inhibit cataract formation?
No, 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 no. This is just, as I said, very, very basic research. When you look at the newspaper headlines, you see how headlines are sort of overblown in the way these people say. Absolutely very, very basic research. There are a lot of more work to be done. But I hear there are some eye drops now that actually kind of use it, but I'm not sure about that. So I won't be able to give you a very clear-cut answer in that sense.
Is there anyone wants to ask any more questions? We still have time. So if anyone, the questions, whatever was written here was asked. So if anyone has any other questions, you may directly ask, ma'am.
Yes, sure. Because I'd like to give a
There,
small break.
so madam, 1 minute. Another question has come up by Dr. Debolina Shorkar. During pregnancy, Down syndrome test only gives probability from certain protein markers. Is it possible to predict definitely?
But that is more of a chromosomal, as far as I know. I wouldn't be able to comment on that. But as far as I know, that is a chromosomal check that has to be done for Down syndrome, where there is one defect in a chromosome. So there are definite markers, and with more and more research in the field, there are definite ways in which even minor changes can be predicted.
Thank you, madam. Another question has come up. Shujata Dhar Datta has asked, is there any hormonal influence for triggering angiogenesis in body?
No, because see, angiogenesis is actually a process that has to occur in wound healing also. Okay, so in wound healing, what happens is blood vessel formation occurs anyway. We have a wound. If I have a cut, there is going to be angiogenesis. And there are other growth factors also, as I mentioned. There is the what is called the vascular endothelial growth factor. There is the transforming growth factor. So these growth factors all put together, it is not just one single reaction. It is a series of reactions that actually occur, a cascade of reactions that actually occur for the angiogenesis to occur, where there is an angiogenin, say, binding protein on the surface of the cells. So this angiogen has to orient properly to sit on the surface. Then there is the nuclear translocation that occurs, and then you will have the process occur. So it is not just one step. There are a whole series of steps, and when you actually do these tests in the laboratory scale, you are not looking at the other setup that occurs in the cell. There are so many other proteins that can interfere, so many other factors that could take place. So that is extremely important in understanding that. So whenever you do in vitro tests, you are not actually mimicking an in vivo environment at all. So when I'm looking at, because I am looking at just the protein, right? So I am looking at just the protein. I am not looking at the cell as a whole. So this is why to look or to translate this kind of research or this kind of ideas takes a lot of time, because you have to look at the cell as a whole, you have to look at the process as a whole, and then find out what is going on. So it's not that easy.
Thank you, madam. Uh, no more questions has come up.
Okay.
So thank you. Just Namrata Ray, one of the participants, has already described, which I was about to say, that you were both a good researcher and a good teacher as well, and
Thank you
all
so
of
much.
us have benefited from the thank yous. I can see here. Couldn't tell you, there are I couldn't count, there are many thank yous. Thank
My
you,
pleasure.
madam.
Thank you. Thank you for giving me this opportunity.
Thank you, madam. We were uh, informed, and we came to know about many new things from your research. Thank you, madam. Thank you
Thank
very much.
thank you. And I'll leave this meeting right now. Thank you.
Okay, thank you, madam. The next speaker has already joined, so we will be starting at 3:30 sharp. So please bear with us for a few minutes.
Subroto, please start.
Hello, am I audible?
Yes, please start the session.
Yes, sir. Welcome to the next session. In this session, the resource person is Professor Tonmoy Pathak. Uh, he is the former professor and head of the Department of Chemistry of IIT Kharagpur. Uh, he has completed his bachelor's and master's from Jadavpur University, Kolkata, and after that, he has done his PhD from Uppsala University, Sweden. He has postdoctoral research work from Southampton and St. Andrews University and University of Karlsruhe. Um, Today, we will be talking about, just a minute. Today, he will be talking about organic reactions in cells and medicinal chemistry. So over to you, sir.
Can you hear me? Am I
Yes, sir.
audible?
Yes, sir. Yes, sir. You are audible.
Okay. Now help me with the technicalities. I have opened my lecture. What should I do after that? Share mode.
Yes, sir.
Sir, there is a video icon at
at
the
the
bottom.
back.
You will find a video icon, and after that there is an icon with up sign, up arrow.
Which is indicating share screen.
Right.
This is the worst part. I used it during COVID and then forgot about it.
Sir, video icon. Can you find
At
the video?
at the bottom.
At the bottom, video icon.
Yes,
After
sir.
that, there is an icon with the up arrow.
Up arrow.
So in the bottom there are first icon is for microphone, second is for camera, and third one is for share screen. First one is for microphone, second one is for camera, and third one is for sharing screen.
Yeah. That I have got. Now I go to
Entire
yes.
screen.
Yeah, I've gone to share screen. Now it's showing me select a tab to share.
Yes, sir. You can select the entire screen. Then the presentation whenever.
Will automatically come there.
Yes, sir.
Entire screen.
So, click on the window which is showing. After selecting entire screen,
Yeah.
then click on the window which is being displaced, and then that option will come, share.
Yeah, I'm doing that. Yes.
Sir, it's coming.
Sir, it's coming now.
So now I'll have to open my slides.
Yes, sir.
Hold on. Yeah, that.
Yes, sir. It is visible now.
Okay, all right. Is it visible?
Yes, sir. Yes, sir. It is visible.
So I'm going to the presentation mode.
Yes, sir.
It's
Now
okay.
It's perfectly fine.
All right. Good afternoon, everybody. I guess it's quite late because we have been listening to several lectures since morning. What I have decided, I will not talk about my own research because this topic is not so common. It's not taught everywhere either. and it's a kind of a mixture of a little bit of biology, little bit of chemistry, and so on. I guess most of you are from biology background. That's the impression I got. Interrupt me, please, if you want. Ask questions. So I will show you some structures and pictures, and show you some reactions as well. But don't be scared, because it's not organic chemistry class or anything like that. So let's see where we go from here. If we talk about medicinal chemistry, it's a mixture of several things. This particular slide is taken from this particular article. You can get it from internet without any problem, and you can have a look on your own what is actually medicinal chemistry. But you can see this picture is quite interesting in the sense the fellow at the bottom is the medicinal chemistry, and he's kind of conducting the orchestra, which is medicinal chemistry. So who are the people involved in it? You can see that structural biologists are there, biochemists, clinical experts, pharmacologists, toxicologists, molecular chemical biologists, cell and microbiologists, computational analytical chemists. And then you see there are lots of molecules which are also dancing to the tunes of the medicinal chemistry chemist. So it requires the input from various disciplines. And if you broaden it further, you can see that it involves inorganic chemistry, chemical biology. Organic chemistry plays a very significant role and very important role. Also the analytical chemistry, cellular system biochemistry, of course, various branches of biology. And what I have not shown is the engineering part of it, because even at the end of the day, when you have a molecule ready to be a drug, how it is to be converted to a tablet, or how it will be put as a solution in an ampule, it will depend on the expertise of chemical engineers and mechanical engineers as well. So all these things put together becomes medicinal chemistry, and everything has to work perfectly to produce one particular drug. This is one of the most conventional diagram that is still valid to a great extent, that what is required for drug discovery. For example, it gives you the timeline. So if you really start with, let's say, 10,000 compounds, and you end up with 250 compounds in the preclinical trial, then 5 compounds eventually reach the clinical trial. From there, eventually only one approved drug reaches the shop where you buy your medicine from. So this is a very critical and very difficult process, and you can see the years, number of years. It takes almost 15 years or so to arrive at one particular molecule. There are a few exceptions though, and it also requires a lot of money. There is no doubt about it. However, if you people are always trying to develop new strategies. This is one of the most recent article you can see from August 2025. The future of pharmaceuticals, artificial intelligence in drug discovery and development. This is also available free in the internet. You can see that. So this artificial intelligence is now being introduced. I cannot say how much of it is used. It will be clear in another 5 years or so. And you can see this it's divided in different parts. I will not go into the detail of it. And you can see the strategic research where your target discovery is required. exploratory research, heat generation, lead identification. That's where you really identify the molecules, big or small. That's a different issue. And then further down the line, you can go for candidate drug selection, lead optimization, that is very important. Then you go to animal models, and then full development, clinical trial, post-approval, etc., etc. And there are various phases of it. So the bottom part is more or less similar to the picture that I've shown in the previous slide, but this one is expected to speed up the process of selection of molecules, and that will see what happens in the near future. This is a picture. I don't know how many of you recognize this particular page. This is taken from the opening pages of this famous biochemistry book, Voigt and Voigt. And this is just the one-page picture of all the reactions that is, or transformations, should I say, taking place in one particular cell. This actually is a map of the major metabolic pathways in a cell. Those who are not familiar with the term, then metabolic pathways are a series of reactions that produce specific products. And each reaction is catalyzed by a distinct enzyme. Now there are 4,000 known, and there are several others, modified ones, which are also being recognized, and so on. This process goes on. Actually, this number continuously changes. What is important here is that one aspect of chemistry, that is physical chemistry, and it is some topics from there like entropy and others. That plays very important role in this particular metabolic pathways, because this requires the whole process requires a continuous influx of free energy to maintain order in a universe meant on maximizing disorder. So just imagine that all these reactions are taking place in a very well-defined way, in one after another, like one product is used for the next step, and so on and so forth. Because if one product is not produced, then one particular next reaction will not take place. So that is an extremely complicated process, and to interfere with the process is also a very complicated task for scientists. But if you zoom in really, then you can really see the molecules at the end, which are extremely important for all these things. And these days, even an organic chemist has to learn a bit of biology. Otherwise, it becomes very difficult to design any molecule for medicinal chemistry. So you can see all these important molecules are there. There is glucose, there is phosphate, pyruvic acid, acetyl coenzyme is very important, citric acid cycle is there, ribose, sugars which forms polysaccharides and so on. You come down to nucleotides, amino acids, and then all this process of nucleic acids, proteins, and all chemical reactions of living things that goes on. And you can see in the middle of it is adenosine triphosphate, which has been highlighted in pink color because it's very important. And this interconversion of organic compounds is extremely critical to the functioning of the cell. This statement is kind of philosophical. You can think that there's no difference between a human being and a bacteria probably, or a cockroach. It is humbling to realize that the same molecules are present in all living things, from the simplest single cell creatures to ourselves. So sometimes they're better in terms of survival than us. That's a different story. But at this point, I just want to highlight, I won't have time to spend much time on it, because this molecule itself, you can spend hours talking about adenosine triphosphate. It is like the gas cylinder in our kitchen. Since you cannot use fire in a cell, so you'll have to go for some different kind of strategies for generating living groups and then particular reactions and so on. This is the central dogma, as you all know. It is simply to highlight that we'll have to fall back on it from time to time whenever it's necessary. So those who had biology in higher secondary, so for them it says this DNA makes DNA like through a process called replication. DNA makes RNA through a process called transcription. You can also have reverse transcription. And then you can have a translation process called translation, which generates protein from RNA. To my students, I have always said that even if you do not do science in life, just remember DNA makes RNA makes protein. Not that molecules get converted into one another. It's a different process. It's a very complex process. But this is very important to remember all this. So for the help of both the chemists and the biologists, a common ground is found that is called bioorganic chemistry, and where organic chemistry is discussed in cells. Okay, all the reactions we study in the 12th standard organic chemistry, how they perform in the cells. So these are the topics which are covered normally in the cell in this book in detail. These proximity effects in organic chemistry, how molecules come close to each other, amino acids and polypeptides, obviously, and their three-dimensional structure, phosphate groups and polynucleotides. Phosphate is an extremely important molecule, and it's the way it gets involved in the hydrolysis and stuff like that. That's a huge scientific study that goes on there. There are certain rules by which you can hydrolyze phosphates. Enzyme chemistry is extremely important. The models for that people prepare. Metal ions, as I mentioned, remember in my one of the earlier slides, that how inorganic chemistry is important in drug designing also. Then comes coenzyme chemistry. And of course, the self-organization and self-assembly, how if you make a long molecule, how it can fold itself into something. Probably, I don't know whether Shagota talked about it in her talk, because she teaches peptide folding and stuff like that in her course in IIT Kharagpur. So the question is now, all right, you have lots of organic molecules. So how many organic molecules are there? So there's a rough estimate. And you know, there are theoreticians who do calculations and so on. So somebody has calculated that there are 166.4 billion molecules corresponding to a defined set of functional groups and atom types. Okay. Then somebody said there are 2.5 million known molecules. 17, if you take 17 atoms, that's a standard estimation of number of molecules in probable drug that you can find. And now the current estimate is that there are 20 million different known organic molecules. The question is then, how do you find an effective drug molecule in that case? This is a $1 million question. I just thought that I would show you this particular slide, although it's not quite connected to directly to medicinal chemistry, but I thought there's a half-mad scientist, Stanley Lloyd Miller. This is just to tell you that everything is connected. You see, in that one slide I showed that you start from carbon dioxide and then you go to various molecules. He did one experiment using different gases into this glass bowl. Here it is glass bowl. And he mixed different gases like methane, ammonia, water, hydrogen, etc., etc. Then he used the spark electrodes, and he used the spark. It is a kind of a prebiotic experiment he did, and then collected the molecules that the mechanism is shown here, the structure is shown here. In 1952, he did this experiment, and it showed that in the mixture that he got from that experiment of gases, he got highly reactive species like amino acids, formaldehyde, HCN, purines, pyrimidines, etc. So this tells you about kind of origin of life. So apparently from non-organic or molecules, you can eventually generate molecules which are important for life. So there is no barrier. So this just a comment while going into the life and its relation to organic chemistry. Um, so those who are a little bit scared about organic reactions that I'll be covering, I can assure you that I will not tell you anything beyond what you have studied in your 12th standard, like nucleophilic bimolecular substitution reaction. Probably some of you studied it in the first year of your whatever BSc or BTech or whatever subject you studied. So there is a nucleophilic substitution reaction, SN2, neighboring group participation, Michael addition reaction, and free radical reaction. So a little bit of these examples will come up while we talk about these reactions, organic reactions in cells. So what is medicinal chemistry? Medicinal chemistry is the science that deals with the discovery or design of the new therapeutic chemicals and their development into useful medicine. And what does it involve? It involves a lot of synthesis of new compounds, then investigations of the relationships between structures of natural as well as synthetic compounds and their biological activities. Now, while we say the synthesis of new compounds, we also mean that we look into the structures of naturally occurring compounds, and then we also modify them. That is also part of that synthesis. This third point is extremely important, this elucidation of the interactions with receptors of various kinds, determination of their absorption, transport, and distribution properties. Like, for example, when you take a tablet, and the process starts the moment you put it on your tongue, and it's the process of molecule breaking down or molecule getting transformed or getting absorbed. The whole process starts there. And studies of the metabolic transformation of these chemicals into other chemicals. That is an extremely important area of research, the last point of it, the studies of the metabolic transformation of these chemicals into other chemicals, because it is quite possible that the molecule as a whole, when you start with it, it's a very good drug molecule, but when it goes into your system, then some reactions takes place, and then something toxic or unwanted material comes out of it. I'll show you one example later on. You can also have, you see, the medicinal chemistry is very interesting in a sense that it has it is very much connected to the history of human civilization. And in fact, each and every molecule that has come up or we use routinely without knowing, there is always a story behind it. Sometimes my students used to get extremely annoyed because they were quite confused whether it was a history class or a chemistry class. But this medicinal chemistry is in its crudest sense was known and practiced for several thousands of years. These Chinese, Indian, South American, Mediterranean cultures have records where the people are using plant extracts. Okay, so about 5,000 years ago, there was a book written by Chinese emperor on herbs. 3rd century BC, opium became known as analgesic, and this Inca civilization of Latin America, these male runners and silver miners, they were using cocaine, coca leaves as stimulant. Greeks used toxins for various reasons and some compounds for sleep inducer. You have heard about the story of hemlock, which was a poison. In India, this Charak Samhita and Shrut Samhita was known, almost 1200 years old. And English translation, those who cannot read Sanskrit can read English translation of it. It is available. When I was working in a company for a small period, we had to look into it from time to time. Not that we could understand everything, but we had to look at it. And if you go back to 1633, a monk, he returned from Central and South America and introduced this bark of cinchona trees to Europe. So if you just look at some of the milestones, very simple one, like quinine, which was isolated in 1820, 1887, first synthetic drug came up, antibiotic drug that came up. 1938, I will talk about this compound a little bit, sulfanilamide group of compounds that came up with it. And Tagamet, this is one of the first multi-million-dollar drug, anti-ulcer drug that was discovered. Then, of course, there was a quantum jump afterwards, and you can take up many molecules as milestones in human history. In medicinal chemistry, but these were some of the earliest ones. Now, what does medicinal chemistry do? Medicinal chemistry, actually, it connects molecules, reactions, and certain things which are apparently unconnectable. In normal human eye, if I place those things next to each other, apparently they don't make any sense. For example, if you take paracetamol, I've taken one of the simplest example possible. And then I talk about liver toxicity, I talk about a molecule called glutathione, and I talk about Michael addition reaction. You see, this organic chemistry has come here, this Michael addition reaction that has come into it. Of course, a lot of chemistry has gone into the synthesis of it, although it looks very simple today, but a lot of reactions were gone into it to make this molecule. But what is important for our talk today is that the Michael addition that is taking place inside the body after somebody consumes this paracetamol. So these are apparently connected, and that's how paracetamol, although it's a very important molecule, it can be very dangerous as well. I'll show you in a moment. Now look at this tripeptide, glutathione. Okay, there is a thiol group here. Okay, this thiol group is here, and this is kind of, it guards our liver. Okay, it's standing there as liver, and in case of oxidative agents, it dimerizes. You can see disulfide bridge, or when necessary, it goes back to the monomeric condition. So this is a combination of these three amino acids, and glutathione is the tripeptide, and depending on the situation, to protect our liver, it either forms this disulfide bridge, or when necessary, it goes back to monomeric tripeptide. So glutathione sacrifices itself if it meets an oxidizing agent through a reversible process. This is very important, that this is a reversible process. It's not like the way we do chemistry in the lab, that once we have done the reaction and got the product, we throw it away. No, nature does not have that luxury. Nature has to reuse the materials for various purposes. So paracetamol, now the question is, paracetamol is a popular and safe analgesic. We know all that. We just buy it and swallow it if used properly. But an overdose is dangerous because irreversible liver damage occurs. Now, what happens is that this is a very oversimplified mechanism that I have shown here so that you can easily follow it. So this paracetamol, once it goes into the system, it goes through an oxidizing agent. And after this, I have shown the arrow, a molecule of water leaves and generates a molecule which is called Michael acceptor. Why? Because look at this double bond, which is flanked by two electron withdrawing groups. Okay, so it's a very highly reactive double bond, and this kind of system is called Michael acceptor. So once this is into your system, glutathione and thiols are extremely highly reactive nucleophiles. Glutathione has no choice but to react with this double bond and form a carbon-sulfur bond. and eventually it aromatizes to form this particular molecule from paracetamol. Now what happens? This molecule is of no use. This molecule SR means this is a tripeptide which is connected to this particular form. It cannot be regenerated from this system. So the system will excrete this molecule from your body, which means for each molecule of this Michael acceptor, one molecule of glutathione will be consumed, and then this will be excreted from your body. So glutathione detoxifies the oxidizing agent, and each molecule of paracetamol consumes one molecule of glutathione. And you'll be surprised to know that there are reasonably high number of cases in the world where people have attempted committing suicide by consuming large amount of paracetamol. It's a very painful death. So I hope no message will go out. But the point is that this is the basic organic reaction that tells you that apparently a very safe molecule like paracetamol can be so toxic because of the fact that a particular organic reaction is taking place inside the body. Okay, so if you have to go for a drug design principle, what will you do? There are several steps, and these steps changes every day depending on the requirement and the availability of new information. Drug discovery without a lead discovery, there are many ways of knowing it from nature or from other sources. Lead modification, you have a molecule, then you modify. You see the word called pharmacophore comes in here. I'll talk about it later. Receptor is there. Where is the molecule going and sitting there, and how is that interaction taking place? Enzyme inhibition and inactivation is there, that I'll also talk about. DNA intercalators, one can really attack directly DNA for with certain drugs, which are also used especially in cancer and related diseases. Drug metabolism, how does it break down? How does it goes into the system, oxidative reactions, reactive reactions, and how does it break down? And another set is very interesting product. You can make a new molecule, but the point is that the molecule has to go into your system through your cell and so on. And not always every molecule is welcome into a body, into any body, into a cell. So this molecule has to be converted into something else to fool those system present in the natural cell, so that the molecule can go inside and generate the drug molecule. Now, it is very important to understand the forces of interactions that bind drugs to their receptors. This is a very, very important area of medicinal chemistry. So there is some theoretical calculation. You don't have to bother about this calculation. So a standard drug was taken with a molecular weight of 200. That was taken. And the number of molecules was calculated by some theoreticians. Okay. So, and they have also calculated that one cell contains about 10 to the power 10 molecules. And this particular drug has roughly 10 to the power 5 drug molecules, which means that for each drug molecule, there are 100,000 molecules of the human body. That means for each drug molecule, you have 1 lakh molecules present in your body. And the molecule will face all of them and then eventually find its target. It's almost like this fellow swimming in a soup without knowing where to go. But it's not quite like that. There are, now the logic comes in, What is the logic? It's easier to use an example to tell you how it really works. So you take this particular molecule, this is a local anesthetic, and it has various different types of structure. If you look at the structure, then you can see that there's a long chain present here. Okay, it's quite lipophilic in nature. Then you have aromatic systems present. It's also kind of a planar system in a way. You have this amide kind of linkage. You have a positive charge, positively charged nitrogen, and so on. So how will this particular molecule find its active site? So there comes these different kinds of recognitions that is present in a particular drug receptor interaction. So what you have, look at this, you have a pyridine ring where you have a charge transfer process that is going on. You have a hydrophobic site. There are hydrophobic cavities in enzymes where the molecule can set in. You have hydrogen bond formation that taking place, ionic or ion dipole interactions. It is there. You can see that ionic interaction is there. And we also have hydrophobic, several other hydrophobic sites. In addition to that, you have several other hydrophobic sites like methyl groups are there, ethyl groups are there. You have a long chain group is there. So these are involved in the binding. The point is, why is it happening? See, it is not very easy. A molecule is just like a child. You ask the child to sit in one place, the child will not stay there, child will go somewhere else. So you'll have to do something so that the child comes back and sit in that particular place. So, and what is that mechanism for a molecule? The entropy does not allow you this organization, but so you'll have to have something else to compensate for that. Now, for each binding, you see, I have given a number. For each hydrogen bonding, that is 3 to 5 kilocalorie per mole energy gain. For each ionic and ion dipole, there's 1 to 7 kilocalorie per mole energy gain. Charge transfer, 1 to 7 kilocalorie per mole. And hydrophobic, for each hydrophobic interaction, single methyl group improves binding by 0.7 kilocalorie per mole. It may look very simple, but when you have lots of hydrophobic interactions, then you can see that several lot of energy gain is there. So, and if you have a covalent bond formation by any chance, then there is a huge energy gain of 40 to 110 kilocalorie per mole, and weak van der Waals forces gives you a gain of 0.5. So this actually altogether, if you do these energy calculations, you can see that this entropy factor is compensated by this huge amount of gain of energy. You have noticed that I have used a negative sign in front of each number, which shows that there's a gain of energy, and therefore the molecule settles in that particular site. Okay, I mentioned the word pharmacophore earlier. Pharmacophore actually is the actual active site of a molecule, which is responsible for the biological activity of that particular molecule. A huge experiment was done using morphine. Morphine is derivative codeine and heroin. So this is the original structure, as you can see, is there. So lots and lots of derivatives of these molecules were synthesized. And you can see I have highlighted them using this blue color. And you can see that molecules they have made, it always showed that some of the a portion of the molecule is highly important for their activity. So there, that is actually the pharmacophore. You can see that the ring was opened up here. This sort of ring was completely opened up. Even then it was showing lots of activities. So from this complex structure, one can come down to this very simplified structure where aromatic ring is required. This six-membered heterocyclic ring is required, and that makes the molecule active. All right. So depending on that, so that is actually the pharmacophore, the most active part of a drug molecule. This helps in a way that let's say that you have got a very complex molecule you obtain from natural sources, but say let's say plant, a bark or whatever. So for a few milligrams, we'll have to probably kill a whole tree. So once you know the structure, once you know the mode of action, then you can really take help from synthetic organic chemists and make derivatives so that you can eventually come down to smallest possible fraction where the molecule still shows its activity. So this sort of modification also leads to new discoveries. For example, very complex structure like this, it's so active that this cannot be given to human beings. It is used for veterinary purposes, where you can see the molecule is 1,000 times more potent than morphine. And this extremely complex structure gives you an idea how you can, if somebody is dependent on morphine, then there is the painkiller activity as well as the addiction part of it. People also try to separate the two, whether it is possible to separate the activities so that the molecule only is used for pain killing rather than the patient becoming dependent on the addiction or the addiction part of it. So this is 10 to 20 times more potent than morphine and very low level of dependence liability. So that's how actually the active part of the molecule is discovered. Now you all know in one way or another that these amino acids eventually leads to the primary structure, secondary structure, alpha helix, beta sheet, etc., etc. For us in this discussion, what is important is the cavity of various enzymes. It looks like rangoli, but you see that there is a very well defined cavity for each of these molecules. You can see that in this particular enzyme there is a white colored molecule which has gone and sat in there, and it's showing its activity in that fashion. We should remember this gentleman in this discussion is G.N. Ramchandra, whose Ramchandra plot is extremely important for deciding on the certain structures of polypeptide chains and so on. We should also remember that there are 6 different families of enzymes. There are various subdivisions in that, but we will not get into this. This, from the name, you can make out the oxidoreductase, oxidation reduction type of reaction. Transferase means it transfers some kind of molecule. Hydrolase means it's hydrolyzes. Isomerases means it's isomerases. Lyases means it just put together things. And ligases, which are also called synthetases, so they also join 2 molecules. So 6 different enzymes are there. Why I'm mentioning it? Because at some point I'll tell you that we were also targeting enzyme for designing drug molecule. This is just a reminder of the structure. People forget the structure over a period of time. So this is just to remind you about this different deoxy nucleic acid and ribonucleic acids. The difference is here, you can see that there is one additional hydroxyl group there shown in red. And one major difference between the 2 series is that in addition to the lack of hydroxyl group here, thymidine is present in DNA, which has a methyl group, and uridine is present in RNA, which does not have a methyl group. We will need this, just remember these 2 structures, we will need this at a later stage. And this is the typical picture, very simplified structure of DNA synthesis, double helix synthesis. As you can see that a triphosphate of a nucleotide, a triphosphate is here, has come closer to the growing DNA chain, and this is being used as a template. The very first central dogma, DNA makes DNA, and this is the template that is being used, and this is the recognition, this is the hydrogen bond. And this Watson-Crick base pairing, this structure needs to be remembered. It is very important that you can see that AT base pairing has 2 hydrogen bonds, and GC base pairing has 3 hydrogen bonds, which are shown here, and these acts as a kind of recognition for picking up a particular nucleotide for the synthesis. But the interesting part is that you can see this hydroxyl group of this growing chain is attacking the triphosphate of the incoming nucleotide. So this particular reaction is actually the nucleophilic reaction that you are seeing inside the cell. Okay, it's a substitution reaction that you are seeing. This oxygen is attacking this phosphorus, and this diphosphate is living as a living group. Okay, so this is the organic reaction that is happening in the cell. Of course, as you understand that I have simplified these things to give you a broader picture. And these two people were responsible for this structural elucidation, and this gentleman died last month, I think. But we should always remember this lady who missed out, unfortunately, who was all there probably before them in terms of determining the double helix structure. But unfortunately, she died very early and missed out on the Nobel Prize. Okay, so Hargobind Khurana's, this genetic code, this is extremely important. The whole picture is giving here. Those who are not so much familiar with the biology can remember this particular structure. It will be very easy to remember that this double helix through transcription goes to initial RNA transcript, and this RNA is then divided into mainly three parts: tRNA, RNA, and messenger RNA. And this messenger RNA is used for the synthesis of peptides using this particular code. And all these reactions, all of them can be explained using typical organic chemistry mechanism and bond formation and so on. It has gone to such a level of molecular level so that it can be explained to now BSc students also with the help of organic chemistry. So these three put together now comes to this central dogma. We'll come back to this kind of picture again and again. So here you have these three ribonucleotides, which are known as codons. They code for one particular amino acid that we have shown you, that these codons are coding for a particular amino acid. And this amino acid synthesis goes on, and this peptide is eventually synthesized. So this gentleman must also be remembered in this particular case. Now there's a small thing, I don't know whether it will show up, I'm not quite sure. Yeah, this is the tRNA bringing in the amino acids. Then this is your mRNA that is there, and you can see this is a growing peptide chain which is coming out. And now this is the whole process of synthesis of the peptide. Now your new peptide has been synthesized. Okay, we'll come to the use later. Now, what is the importance of chirality? I'm sure all of you have studied chirality, the R and S version of molecules. Now, this is a very well-known molecule. It's called thalidomide tragedy. This molecule was actually used approved as a sedative in Europe in 1956. And pregnant women were given this particular drug first 3 months of pregnancy, but they gave birth to babies with birth defects, and it was withdrawn in 1961 after close to 10,000 babies around the world were born. As I told you, remember in one of my previous slides, the moment you put a molecule on the tongue or inject in the body, the molecule starts undergoing transformation. So this molecule is an example of how much careful one has to be about this transformation of molecules into the body. So the dextrorotatory isomer of this is the chirality carbon here. Dextrorotatory isomer is sedative, but this, once it goes into the system, it goes through once again an organic reaction, ketoenol tautomerism. You see, this is a keto group, and it is undergoing this ketoenol tautomerism here. And once it goes back again in its keto form, you have a complete scrambling of the center. That means even if you start with the pure dextrorotatory isomer in your body, it will be completely one-to-one mixture of dextrorotatory and levorotatory. And this levorotatory isomer cause this birth defects, and these are the babies known for the birth defects caused by the thalidomide tragedy. So drug companies have to be extremely careful. Also, After so many years, in 2012, this particular company apologized for the victims of thalidomide. As I was telling you, that all molecule has a history behind it, whether it's a good one or a bad one. History, nevertheless. Okay. So the receptor chirality, when you see, then proteins are chiral substances, and two complex formed between a receptor and two enantiomers are diastereomers, and as a result have different energies and chemical properties. So in a simplified version, I've just taken one example, that you can see that this particular receptor has three recognition sites, all right? Now, one receptor site recognizes aromatic ring, one recognizes the hydroxyl group through hydrogen bonding, and the other one has a negative charge so that it can recognize the positive charge. Now you can see that in this particular receptor, okay, this particular receptor, this hydrogen bond formation is taking place, and minimum three recognitions are there. But in this particular case, only two recognitions are there because of the chirality of this particular structure, or the stereochemistry of this structure. Or if you have problem with these two terms, just use the word the disposition of various functional groups in this particular carbon. If this hydroxyl group does not reach to that particular to that particular site, which means that this R-epinephrine will be more active a molecule for this particular receptor, and S-epinephrine will not show activity. If it is a neutral kind of molecule, in the sense that it will not show any molecule, then it is fine. But if it is it has some side effects, as I shown you in the earlier slide, then that can cause a lot of damage. For example, this for allergy, the cetirizine tablet that you buy, it's a mixture of R and S, but one of them is good for you, other one is of no use. Okay, so there is no problem. But if you go to buy levocetirizine, you will see it becomes more expensive because the separation and synthesis of that particular stereoisomer makes it more expensive. So enzyme as a target is a very important area of research. Why? Because enzymes are easy to isolate and purify. It can be crystallized. It can be co-crystallized with the substrate, and that way it can reveal the binding pattern. Now, if you look at this particular reaction, this is the biologist present in the crowd will know that this particular reaction is so very important. The phosphorylation from glucose to T-glucose 6-phosphate. Okay, this whole process of glycolysis depend on this particular transformation. The point is very interesting. Now, this is the chemistry part of it. If you look at the cell, typical cell, there is 70% water, and the rest are inorganic ions. You have a little bit of this. But the point is that you have 70% water. Now, when you are doing a reaction where a molecule of water will be generated, this reaction cannot proceed in a soup which is made of 70% water, because the reaction will be always pushed backward and glucose will never be phosphorylated. So this is a puzzle for an organic chemist. What happens? The biochemist knows it very well. What happens is that this adenosine triphosphate, which I have shown you earlier, comes into the picture, and it goes through this particular reaction. It cannot, like this, it cannot go here and do this organic chemistry type of reaction. See this hydroxy group, you are using it as a nucleophile, and if you are reacting here, okay, so this will not give you this kind of system on its own. So what is required here? You need an enzyme. All right, so this is the hexokinase enzyme which is coming in. The glucose is coming into the system and coming into this kind of system there, and the reaction is taking place inside the cavity of this particular reaction. So by mixing this one and adenosine triphosphate in a solution, you will never get the 6-phosphate of D-glucose. You will have to have an active site cleft, which is shown in this particular enzyme. Okay, so this cleft I have already showed you. The cavity is extremely important for this kind of reaction to take place. Now, those who are not so familiar with this enzyme inhibition type of reaction, that's how it works. That if you have a substrate, this is the natural substrate, this is an enzyme. Think about a football, it will be easier. So this has a particular size, so it goes in there and then it gets transformed into something else. This is the natural reaction that is taking place. And inhibition happens when a similar looking molecule, which looks almost like that, but it's not exactly the same, comes here, sit in this active site, and does not allow the natural substrate to come in. That means this molecule is competing with this one and inhibiting the activity of this particular enzyme. So you can look at this picture and remember it as a simplified picture of the competitive inhibitor of a reaction. You can have different types of it. I'm not going to it, but just remember there is something called allosteric inhibition. Allosteric means that it does not go to that active site. It goes and sits somewhere else and distorts the enzyme in such a way that the substrate cannot come into the active site. Okay, so allosteric inhibitor are extremely difficult, if not impossible, to design. So people always go for this sort of inhibition where they know the structure of the natural substrate and therefore they work on it to make a modification of it. In fact, which I have not mentioned here today, the previous speaker, Professor Shagota Dasgupta, and I, we together worked on a project called ribonuclease inhibition. I used to make lots of molecules of it, and then we used to have joint student. We'll go to her lab and do the study on the inhibition. So that's a different story altogether. All right, now let's look at we have looked at the toxicity of a molecule. Now look at one molecule, one of the best known molecule in the literature, or in the use, in terms of its use, aspirin. Once again, you can see that there are 4 apparently unrelated molecules: aspirin, prostaglandin, cyclooxygenase, arachidonic acid. Okay, so they have to be brought together. It's a very difficult task. If you think that once the answer is known, it's very easy. But if you ask somebody to search in the dark and find out the connection of a molecule with its various other things with which it interacts, then it's a very difficult process. Okay, so let's see what happens here. See, arachidonic acid, for example, this one, is a polyunsaturated fatty acid present in membranes of the body cells and is abundant in the brain, muscle, and liver. So what happens is this arachidonic acid undergoes a transformation with the help of an enzyme called cyclooxygenase, or it's called prostaglandin synthase. All right, so you can see that C13 carbon, there is a reaction that is taking place here to form a stable conjugate radical. I told you that one of the 4 reactions that I'll be talking about, organic chemistry in cell, is this radical reaction that is taking place here. And this is forming this 5-membered ring, and which eventually generates prostaglandin and many other compounds from there. Now, what was known in the literature that inhibiting prostaglandin synthesis can bring about a reduction in inflammation and pain. That was known. So some people connected these apparently unknown things together. And this is a very simple, very, very simple experiment done in 1970s, where absolutely nothing was known, that people made 2 different types of aspirins, where this methyl group was tritiated. It's a tritium, so it could the radioactivity can be followed, and a benzene ring was tritiated, and then they incubated it with the cyclooxygenase and so on and so forth. That they eventually isolated this dipeptide and observe that the serine, now you need to see why I keep talking about the structure of molecules, is that one needs to know the structure of serine. Serine has one CH2OH group present there, and that's why it becomes so important. So they have, once they have done that incubation, they got that serine molecule and saw that this serine molecule has been acetylated. You can see that one. This hydroxyl group of serine present in cyclooxygenase has been acetylated. So through this acetylation, the cyclooxygenase enzyme has been inhibited. So this is your serine with CH2OH, which is present in cyclooxygenase, and this hydroxyl group is acetylated by aspirin through this transesterification reaction, okay, organic reaction in cell. This transesterification acetylates the enzyme, and therefore this enzyme cannot synthesize these prostaglandins anymore, which were responsible for the pain and inflammation. And apparently now it looks very simple, and this gentleman in 1982 was awarded Nobel Prize for this discovery. So you can see that how easy it is to get Nobel Prize once you are working in the right kind of problem. This is just to mention the name of this gentleman in this discussion, Upendranath Brahmachari. Probably not my word, he is probably the last medicinal chemist of India, because after him, no Indian in India has discovered any medicine except for one molecule that has come up in the last 1 or 2 years in a company. Okay, so this urea stale bean mechanism has also been studied in IICV Kolkata, that it's a topoisomerase inhibitor, but I will not go into the detail of it. Okay, coenzymes as target. I'm not sure how much time do I have. How long can I keep talking? I have time, I guess. Will the moderator inform me, please?
Sir, there are almost 15 to 20 more minutes.
Oh, lovely. Thank you. Okay, so these coenzymes, which are actually derived from vitamins, it's a fantastic area, especially when you teach in a class with these structures. It's quite a fascinating area. I have picked up only one or two of them. This folic acid is a very important molecule, and you can see that you can get tetrahydrofolate. You can see these double bonds are hydrogenated here. Tetrahydrofolates are formed. These are one set, and the other one is this pyridoxal phosphate. These two are I have picked up just to show you some reactions, important reactions. Now, those who are interested in biotransformations, for example, one can use these pyridoxal PLP-dependent enzymes. I have done this work during my postdoctoral studies. You can use this one in a flask, and by properly maintaining the temperature and pH, you can use these molecules in biotransformations for making new molecules, just like chemical reaction. Instead of chemicals, you can use this. These are very easily available. Okay, look at this particular reaction. Just note one particular stage. This is the biosynthesis of bacterial dihydrofolate. Since we cannot we cannot use synthesize vitamins. We'll have to get it from other sources, but bacteria can synthesize its vitamin. So this is the biosynthesis of bacterial dihydrofolic acid. You can see that this is the starting material. Don't bother about the name and the structure. Just note this CH2OH group at the end of this tail. Okay, so this phosphorylation is taking place here using adenosine triphosphate. Phosphorylation is taking place. What is important here is that in the biological system, phosphate also works as a very good leaving group. The way you use a bromo compound or a tosylate and so on. So a reaction takes place at this stage. Para-aminobenzoic acid, or the biologists will remember it as PABA more easily. This dihydroteroid synthase does a reaction and forms this dihydrofolate. Okay, so from there, the bacteria synthesizes its dihydrofolic acid and so on. It synthesizes. But this step is extremely important. Remember this one once again, that this dihydroteroid synthase, this para-aminobenzoic acid is coming in and forming this dihydrofolate. So this bond formation is extremely important for the synthesis of the folic acid and the survival of the bacteria. Now, if you look at the next step of the biosynthesis of bacterial tetrahydrofolate, it involves this dihydrofolate reductase, right? And you have NADH here, which supplies the hydride here. And it goes in a stepwise fashion. You don't have to remember the complexity of this reaction. Just remember this hydride is being supplied here to reduce this double bond. Another molecule of another hydride comes in to reduce this double bond. And so you get this particular molecule, this dihydrofolate you get. And from there, I have picked up two different tetrahydrofolate based coenzymes. Okay, so humans do not synthesize vitamins, need supply from external sources. Bacteria synthesize their own vitamins. So now you have two parallel pathways in the system. So in a human body, if there's an attack by a bacteria, then you can attack that particular route without affecting the route that humans use for the synthesis of vitamins. And there, these two tetrahydrofolate based coenzymes I have picked up as an example. So there are 6 different tetrahydrofolates. I have picked up only 2. Remember, what is the difference? The only difference is that this is part of a ring shown in a pink color. This is shown here, and there is an aldehyde group here. So these two will be useful for our reactions. Okay. Now this is the time of Second World War, or kind of related time. And yeah, it's a fantastic story. A molecule called prontosil, you can see this structure here. A molecule, it was a dye factory, it's a German dye factory. This gentleman was named Tomac. In only 31, Tomac tested the compound in mice that was having some bacterial infection, and that was found that it was effective against bacteria. So this prontosil was effective against bacteria. So the story goes, okay, I'll come to a minute. The story goes that his daughter cut her finger. And at that point of time, in 1930s, there was no antibacterial therapy, nothing whatsoever was available, and she was about to die. So in desperation, Domac injected her daughter with prontosil, and the daughter survived. Probably that was an example of the first human trial in the history of medicine. After that, thousands of sulfonamides were synthesized. First structure-activity relationship was done, and then new lead compounds from. Actually, this is a typical classroom topic where you can really make people aware of the relationship between structure and activity of molecules and how modifications are done depending on the activities. Domac was awarded Nobel Prize in 1939 for physiology and medicine, but he was a German, so Hitler did not allow him to go to receive the award. So after Second World War, Nomak went to receive his award. But penicillin was already discovered. This is the unique thing. Penicillin was already discovered, but penicillin was for some reason was not marketed. Okay, so this period of Second World War, people were thoroughly treated with this sulfonamide bacteria, antibacterial compound. Now this is just a comparison. I asked you to remember this particular step of the folate synthesis, this para-aminobenzoic acid, and this was the intermediate for the bacterial folate synthesis. Now see what is happening here. This sulfonamide, which is a breakdown product of brontosil, it compares exactly well with the para-aminobenzoic acid. So it goes in there, does exactly the same reaction, forms it, but since it does not have any more carboxylic group, so therefore further amide bond formation cannot take place, and therefore the bacteria cannot have dihydrofolic acid anymore. And if you cannot have dihydrofolic acid, what happens? I'll tell you in a moment. But here is the comparison. It almost matches, by chance, it matches at the angstrom level, the molecules match, and therefore it is very easy to fool each other, the enzyme. So therefore, these dihydrotoluene synthesis can be fooled very easily, and when you have large amount of this molecule given as tablet from outside, so it will outnumber the naturally occurring para-amino benzoic acid, and therefore it will show its activity as an antibacterial compound. So here the bacterial thymidine synthesis is stopped. I will not talk about penicillin. Penicillin is a better known story, so I leave it for some other time if I get the opportunity. DNA target is what I wanted to say. Mustard gas used to be poor man's nuclear bomb. Actually, we have generated many ways of killing people. So mustard gas used to be very efficient one. Now some of you will not like this picture, but I think sometimes it's better to see it. This, if you have little bit of mustard gas on your body, if you have more of it, and if you have huge number of people exposed to mustard gas, it was done in during First World War. This will cause horrible death. This is the simple molecule. What happens? Just nothing. There are two chlorines and one sulfur in between. See, I talked about neighboring group participation in one of my slides, the 4 reactions, organic reaction that I do. So what happens is that when you have a sulfur, sulfur actually participates and generates a positively charged sulfur, which is a very efficient leaving group, and then the next nucleophile can come in and attack here. Now, I have shown this example with one reactive arm, okay? So what will happen when you have 2 reactive arms? So you have 2 reactive arms means that this guanine residue of DNA, this is the highly reactive part of guanine. And this nitrogen will attack this carbon, this bond will break. Next reaction will take place with another to open up this particular day. Now you can see that this second arm is also ready to undergo reaction once again. Another thing has also happened at the same time. Since this nitrogen lone pair participated, this nitrogen has become positively charged, which has made this glycosidic bond extremely used and breakable, even with the attack of a small molecule of water. So what happens here? So one part of a DNA, one DNA guanosine, deoxyguanosine is reacting here, forming this. This is undergoing an intramolecular reaction, forming another nucleophile inside. So now a deoxyguanosine from the another helix of the double helix comes in and reacts here. So two deoxyguanosines of the double helix of a DNA is now locked through a covalent bond. So what happens there? That means if you go back to the central dogma, once again, the DNA goes through a replication to make DNA, and then the whole process starts, is stopped at this stage. Why? Because for this DNA to make DNA, the first part is this unwinding of DNA. This double helix has to separate themselves, okay, and then the arms will eventually get involved into the daughter DNA synthesis. So unwinding is required for new DNA synthesis. What is happening there then, the moment you have I'll leave this structure for a while. So you have a deoxyguanosine in this arm and a deoxyguanosine in this arm. They are now connected through a covalent bond. That means they cannot separate anymore. That means you cannot have the unwinding of DNA for the synthesis of DNA. Therefore, DNA to RNA to peptide, the whole process is stopped. Now, lots of scientists looked into these reactions thoroughly and came up with this molecule. This is nitrogen, not sulfur. Sulfur can stabilize its positive charge very well, very efficiently. But nitrogen, it can stabilize positive charge to some extent, not as good as sulfur. And at the same time, since it's a trivalent atom, you can have another group attached to it, by which you can either pull that lone pair of electrons from it, or you can push it into it, making it sometimes more reactive, sometimes less reactive. If you go back to your very early 12th standard kind of chemistry, you will see comparison of pKs on the basis of availability of lone pair of nitrogen. Then you will see what I'm talking about. So what happens now? This whole process is repeated. So instead of sulfur, you have now nitrogen sitting here. The same reaction is going on, but it's going on under a much more controlled reaction. Okay, you are really in a position to manipulate the effectiveness of this intramolecular reaction. So it's again the same reaction, the another DG from another helix is coming in and forming this bond. So this double helix is now once again bound by this nitrogen. And what is happening because of that? It means that it's like a ladder. You have this once original ladder with black. Now you have added more of these steps, these blue ones, into that ladder, which is making that ladder stronger. And you are not allowing this ladder to unwind what was happening there so very easily. And how does it help? It helps because in cancerous cells, you are stopping the unwinding of cancerous DNA. And therefore, you are coming up with an anti-cancer molecule. And this is the compound called chlorambucil. This chlorambucil is an anti-cancer drug. It is used, it's called nitrogen mustard, and it is used even today. And this knowledge is generated from the chemistry of a deadly poison, which led to the development of a medicine for treating cancer. Okay, I will jump this one. This is the HIV. I wanted to mention that in one connection. Look at this. This picture I have already shown you. Now, this one is the nucleophilic displacement that is taking place here. Now, look at this compound. When there was nothing, early 1980s, people were dying all over the world of HIV infection. And this molecule was very quickly brought to the market, knowing the toxicity of the material and so on. But the point was very interesting, that if this process can recognize this molecule, you see, nothing much has been changed. Thymidine remains thymidine except the hydroxyl group. Here, the azide group has been incorporated. So if you can make the triphosphate of this one, then the triphosphate of azidothymidine will go in there, form the bond, and since there is an azido group and no hydroxyl group, then the chain termination will take place. And it did work very well. Even today, this molecule is in use for the treatment of HIV, but it is used in a cocktail drug, it is called these days. Okay. One more example I'll give you is fluorouracil, which is an anti-cancer drug. Once the same route, deoxy uridine is made, where this fluorouracil is incorporated and the triphosphate is made out of it. Here, the thymidylate synthase that is involved, which is very important because in nature, deoxyuridine gets converted into thymidine because this thymidine is a very precious material and it goes through these kind of steps. And you remember this particular structure, I said that I will show you, tetrahydrofolate. This particular carbon is one carbon that is transferred from tetrahydrofolate to deoxyuridine. And this whole process goes on like this. Just remember this step. Look at first step here, nitrogen CH2 carbon bond formation is taking place. And this hydrogen is picked up and an olefin is generated. Here, once again, an organic reaction, Michael addition and hydride addition is taking place. And this addition is generating a methyl group here, which is eventually giving you the thymidine. That means from deoxyuridine, you are making thymidine. People took advantage of this particular reaction. And what they did, just look carefully. All my hydrogens will disappear and the fluorine will appear there. Yeah. So all the hydrogens have disappeared and the fluorine has appeared here. So fluorine is there, fluorine is there, fluorine is there. So the first step works, no problem. Enzyme does not understand. Second step also works. Enzyme does not understand. And nitrogen carbon bond formation takes place. Now see, this is a covalent bond formation that taken place. The next step, if you remember, previously this hydrogen was picked up. This process cannot go on anymore because this fluorine cannot be picked up. So all these things are now kind of a suicide inhibitor. So the enzyme, the coenzyme, the substrate, everything is killing this particular enzyme. So in the cancerous cell, this thymidylate synthase is non-functional and therefore the methylation cannot take place and therefore the synthesis of cancerous DNA is stopped. So this is actually the step where the difference is taking place. In this case, hydrogen can be picked up and methyl formation can go on. And in this case, fluorine cannot be picked up and therefore fluorouracil kills this particular enzyme along with the coenzyme. Okay, we have come very close to the end of my talk now. As I said that there are something called prodrugs, because you have made a molecule does not necessarily mean that it will work into your cell, because there are also efflux pumps and stuff like that which will throw out the molecule from the system. The question is about the bioavailability, for example. Is it going there? How stable it is? How stable is it? You see, when R is equal to H, it undergoes an extensive breakdown, okay, when given orally. So this is esterified. It is converted into some kind of a ester. So this is stabilized. It goes into the system and then eventually the ester bond is broken. Then the molecule goes in there. Solubility is a problem. It's a steroid molecule. Then it cannot mix with water because your blood will be the carrier of the thing. And so therefore you make the phosphate of it. So it becomes a very charged molecule, becomes specific soluble in water, and then it can be used. The site specificity is a very important area. We could not cover it today. This blood-brain barrier is a very important issue for treating anything in the problem in the brain. So you have to design a carrier molecule in such a way that it goes through the membrane, and then the oxidation takes place and retains is inside after it crosses the blood-brain barrier, and only after that the main molecule, drug molecule here, is released. Absorption, distribution is a very important area. It's an anti-glaucoma kind of compound. The molecule with the free hydroxyl group does not work. This bivalveol derivative, it's also an diester that is given there. And after the drop is given, you have lots of esterases molecule. Esterase is an enzyme which chops off the ester bond, and then the molecule shows its activity. So prolonged release, it is also a very important area of research, that once you give a molecule, then it will go on releasing for a very long period of time, helping the patient so that the patient doesn't have to take too often the medicine. Poor patient acceptability. This is quite an interesting one. This is given to the children, for example. And the one with free NH is extremely bitter in taste, and it's almost, you know how difficult it is to feed babies with bitter medicine. So this ester was actually made, an acetyl ester, and then it becomes completely tasteless, and then you mix it with some kind of a banana scent or whatever is available, and the molecule, after going into the system, this bond is clipped, and then it shows its activity. You can reduce the toxicity also by doing ester. Formulation problem, I don't know how many of you recognize this particular molecule, because in some cases formaldehyde, which is a gas, can be used for treatment. So this compound is made with ammonia, and only under pH, acidic pH, the molecule breaks down into formaldehyde and ammonia. This compound is used as a urinary tract antiseptic. So there are many ways of dodging the system and so on. I have shown you this list. You can have various modifications in the system: oxidative activation, reductive activation, nucleotide activation. You can attach two molecules together and so on. Okay, I showed you the examples of prodrug. This is not a mythology. You can see many of you were treated with this particular medicine. You can see that there is a very simple ester bond formation. Okay, it's a penicillin derivative you have here, and the esterases cleave it. Esterases cleave it, and you generate this ampicillin molecule and this acid sulfone. This one is not the drug actually. This one protects this molecule, okay, because of where you have this bacterial resistance and so on. There, this molecule does the work, and this one protects this one from the breakdown by bacteria. This one is even more common, augmentin. I'm sure everybody in the audience has consumed this particular molecule times. It also works more or less in the similar fashion. So this is amoxicillin. This is the antibiotic that is original works, and clavulanic acid is a non-antibiotic beta-lactamase inhibitor. So beta-lactamase inhibitor, by inhibiting that beta-lactamase, it allows this particular molecule to show its activity. There are two different ways of handling the problem. One area has really come up recently that you can reuse. You can have a relook at the molecule and whether it can be used for new purposes. I just mentioned one, this thalidomide. Remember, the material I said was dangerous, but then the people came up with some other ideas that, okay, fine, it's not be given to the pregnant women and so on, whether we can use it for some other anti-cancer activities. Aspirin is a wonder molecule. God knows people should have a stock of aspirin all the time at home for various reasons. Every now and then, people come up with new activities of aspirin, and so there's a huge list of molecule you can see. I just mentioned these two slides because we still have not forgotten the horror of COVID. So this is one molecule that came up with it. I will not go into the detail of it, and it shows how it binds to the active site, the main protease actually, and shows its activity. Lots of new molecules came up. If you remember, I don't know whether you remember, Remivir there was a huge black market of Remivir. At that point of time, some people came up with the idea that it will solve all the problems and so on. Just partly true. But all these new molecules and some old molecules, people had a new look at it, and they came up with all the new types. And always, new molecules are coming up, but neither the synthetic chemists nor the biologists can do it alone. They will have to hold hands together, come to a common ground, and find out how the things work, and it will go forward. So it's a long way. Sometimes it reaches its destination, and sometimes it does not. But where does this terminate? Where is that? It's in the end. All this craving, all this striving, where do they culminate? It's all for the good of the people. People are coming in. There are some toxic things, of course, side effects are there, etc., there. But people are really trying hard. I know many of my students are in various companies. They are given the task of making sometimes hundreds of molecules, the derivatives of the same thing, because that whole bunch of derivatives will be sent somewhere else, and they will look at their activities. One methyl group there, one fluoride there, one chloro there, and so on. It's a tedious job, and I hope Indians will also jump into this area, which is for the help of the humanity. Thank you very much for your kind attention.
Thank you, sir. Thank you, Professor Pathak. And thank you for this wonderful session, this wonderful lecture on this interesting topic. And now there is a query in the chat box from Dr. Namrata Ray that protonsil was a prodrug actually, got transformed from the sulfonamide inside the body, right, sir?
Yes, sulfonilamide. Yes, that's right.
Then there is another query from Manoshi Mondal. Prontosil saved Domag's daughter. Is it work directly on the isolated bacteria?
No, she was injected directly. It was the first human trial probably in the history of medicinal chemistry, because he had no other option at that point of time. There was nothing available, so he injected it.
Okay, thank you, sir.
And Obhijit Vishwas and Annesha Chaudhuri have raised their hands. Please. So I ask first Obhijit Vishwas.
Yeah, thank you, sir. Thank you, sir. Sir, you have covered a lot of things, starting from the very basic things. I have a general question: is that recently, sir, nanomaterials are used as a drug delivery system. So, how much they are safe for the human?
Yeah, you see, I whenever I have the opportunity, I ask this question. That now it seems that people have come up with this terminology of nanotoxicity. I must be very careful, and I must be very candid to tell you that so far I have not got any clear-cut answer to these particular questions. This is on an academic scale. This is fine, and just to mention a point that my elder son is an immunologist, and he is doing a postdoc where he is given lots of nanomaterials, and he knows how to cut these rats, poor rats. So he tells Baba, I just inject them with those black mass, and this poor rat dies. But some activities are there. Okay. Now I should not denigrate this because I am not quite sure, honestly, whether those who are working with nanomaterials, how much they are knowledgeable about the toxicity part of it. You see, somebody has to be knowledgeable both the nanomaterial as well as how does it contribute to the toxicity in a natural system. So I honestly almost, but I have retired almost 1.5 years. But I try to keep track of things. I didn't get a very clear-cut answer to these particular questions. To be very frank, no, I don't have an answer. If you have any answer, please send me one article.
Yeah. Thank you, sir. Yes, actually, also I have also faced the same difficulties like that carbon nanodots. Recently, people are using some of the people saying that this is the very good for the as a drug delivery, or some of their cytotoxicity is much more, so that it cannot be used. So very
Yeah,
confusing.
the other issue is what I do not get a clear picture is that I am delivering a nanomaterial. Now, after going into your system, is it still remaining nano? Or is it falling back to the, because if you really look at this ratio of one molecule facing 100,000 biomolecules, then does a nano remain nano throughout its life in such a biological soup? I do not have that answer. I have asked this question in several occasions. I also did not get any clear-cut answer. However, the research must go on, because only after crossing certain unknown areas we'll be able to find an answer to that particular question.
Thank you, sir.
Welcome.
Thank you, sir. There is another questions from Onnisha Chaudhuri. So I
Yeah.
request Onnisha Chaudhuri.
Thank you, sir, for your very nice presentation. It's really very interesting. Sir, I have one query. As you have mentioned, Prontosil is a very good antibacterial agent. Can you please let me know which pathway or machinery of the bacterial system is targeted by this drug?
Okay. Prontosil is not anymore. Okay.
Okay. Okay.
At that point of time, it was very good. The pathway I have also shown you. If you remember this one, if you remember that PABA was attacking, para-amino benzoic acid was attacking.
Yes.
Now, if you look at the breakdown product of Prontosil,
Yes.
okay, you also have amino group at the para position, and
Yeah, yes, sir.
okay, so in the
Mm-hmm.
para position you have SO2 NH2. So if you compare that structure with para-amino benzoic acid, then you can see that para-amino benzoic acid, after forming that bond, that carboxylic group was available for the synthesis of folic acid in bacteria. But the
Okay.
moment you dump this sulfonilic acid into it, this SO2 NH2 cannot form this peptide bond anymore. So this bacteria is
That is the drug.
starved. It starved of folic acid. Actually,
Okay.
folic acid, if you remember, I have shown you at the end that it delivers one carbon molecule, which is extremely important for the synthesis of deoxyuridine to thymidine. So this whole pathway
Means
is
bacteria is
completely
deprived of nutrition. Yes.
nutrition will be the terminology. I don't know what is your background, but my terminology is that it is deprived of one carbon atom.
Okay. 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 Right.
Okay.
Thank you, sir. Thank you.
Welcome.
Thank you.
Now I request Dr. Namrata Ray.
Please.
Sir, thanks a lot. It's great to listen to you always. This is the umpteenth number of time that I'm listening to you, and it is
Oh
as
my
usual
God.
very informative. Very informative and very engaging. Sir, because I myself teach medicinal chemistry to my students. So sir, one thing that I wanted to know is I have not found in books. You talked about combination of synergistic drugs, like in sulfamycin, right, sir?
Also augmentin is there, which is much
Right.
more common, you can say.
Right. So, sir, co-triboxazole is also a combination drug
Yeah.
where the sulfur drug first inhibits dihydropteroid synthesis, and then later on the dihydrofolate synthesis. Trimethoprim does that. So,
Okay.
sir, is there a combination of drugs where both independently might work on certain biosynthetic pathways, but when they are combined, one actually inhibits the function of the other? Is there any such example?
You see, this problem can be handled in many ways. See, what happens is that you have, let's say, enzyme 1 giving you compound X, and then enzyme 2 coming in giving you Y. All right. So you can target enzyme 1 and enzyme 2. All right. But sometimes there are overlapping cases. Okay. Two circles, for example, are overlapping. Okay. So in that case, you can use two completely different molecules which can come in and act together. In fact, in some, I don't know what book do you follow, but in some books this has been discussed in detail, that how it works in the number of bacteria. If you go count the number of bacteria diminishing by this, it works very well depending on that particular problem. The problem is of talking about medicinal chemistry, the question slowly starts moving towards the answer that only a doctor can give, actually. Under what condition, what can be used. Okay. So depending on the situation, you can design your drug. See, these things don't fall from the sky. People look at the problem. One molecule has reached its limit, and there is nothing available to replace it. Like this HIV, for example, I talk about, this cocktail drug that has been used. If you look at the whole HIV cycle, there are several enzymes involved into it. So you can go for this azidothiamidine, for example, okay, which is a reverse transcriptase inhibitors, and you can also use another one molecule which is a protease inhibitor. So it is going somewhere else and still working together. It's killing in two different places. So that is also another form of using synergistic drug. Each and every problem in medicinal chemistry is a separate problem. You cannot really use this. Some knowledge you can use, of course, but you cannot use this particular knowledge and transfer it into that one. So I could not answer your particular question. If you have any specific query, you please feel free to send me a mail. It is available in my that abstract. That mail is available. I'll be very happy to answer if I can to your question. So if you can be specific, I'll try to give you an answer.
Okay, thank you, sir.
Okay, thank you, sir. Thank you, Professor Pathak.
Okay, so thank you very much for giving me this opportunity. Actually, but I somehow like to speak directly rather than through the screen and all that kind of thing. It will be nice to see faces while talking. That I feel some better. All right. So thank you so much for giving me this opportunity. All right.
Thank you, sir.
Have a nice day. Yeah. Have a nice day.
Thank you so much.
Okay.
Thank you, sir.
Bye.
Thank you, sir.
Bye.
Thank you, sir.
Hello. So we are closing the session for today. We will again meet tomorrow at 10:15 a.m. for the sessions of day 3. Thank you all.
Okay, ma'am. Thank you.
Thank you, ma'am.
Thank you, ma'am. Thank you.
Thank you, ma'am.
Madam, may I question?
Yes, please go ahead.